also the consultant nations, which is a very big thing for us. And also we are fortunate to have 2 stations there, Maitri and Bharti. Oh, sorry. Maitri and Bharti stations. And on these 2 stations, our Indian scientists visit twice, actually once in a year. One summer expedition remains for 3 months and winter expedition remains for 10 to 12 months. And these expeditions are conducted by NCOR, National Center for Ocean and Polar Research, Goa, under Ministry of Earth Sciences. So we have to have collaborative work with them so that we can conduct our studies. It's also called as Antarctica is a natural laboratory, and it mimics the conditions like space. And science which is being done is on earth science, life sciences, space sciences, environmental sciences, because of its challenging conditions. And these challenging conditions lead to various diseases, be it environmental or the individual problems. Isolation, fear, radiations, many problems are there which people face, but still it's adventurous, and how to reach there is also a big thing. Now, how to reach is by ship or by plane, but generally the expeditions are being done by ship, and it is seen that when the ship crosses the 40-degree latitude, it is so fiery, there is rolling and pitching and people start chanting Hanuman Chalisa. So it's so difficult and fearful. So that puts a lot of pressure on the human body, and it changes the, there are changes of series of physiological and psychological changes. To study all these changes, various life sciences laboratories visit there and do science. One of them is DIPAS, Defenses of Physiology and Life Sciences in DRDO. We have conducted 14 such expeditions, and last 4 expeditions were led by me, where we found out that what are the effects, what is the impact of these environmental conditions, Antarctic stressful environmental condition on the immune system of the body. So we collaborated with NCOR, Ministry of Earth Sciences, and our project was through for conducting experiments on immune system. We were with them right from the beginning. How candidates or the expedition members are selected is based on some criteria. They are sent to All India Institute of Medical Sciences, Delhi, for their medical checkup, and those who are found fit are sent for mountaineering training at Oli, Uttarakhand. After that, then they start going to Goa, and from Goa, then they go on to Cape Town, from where they board the ship and start their journey to Antarctica. So we have collected the samples at various time points so that we have a longitudinal study. Right from the beginning, we were with them at Delhi when they came for the medical checkup. And then at various time points, we collected saliva, stool, urine samples. And collecting stool samples was also very difficult because, like as morning it was said that people, the person who collects the samples is being isolated and not many people mingle with him. Anyway, that's one of the challenges. So we started collecting demographic data, various aspects were collected, and started conducting the experiments. After we received the samples from Antarctica, because there was no possibility of doing experiments there, we collected samples, brought them here, and we did various parameters, where we found that there was increase in pro-inflammatory cytokines, indicating Th1 biased immunity, and in both short as well as long exposure studies, that is summer and winter expeditions both. There was decreased levels of anti-inflammatory cytokines and interestingly increase in IgA levels. And later on we also did IgA1 and A2, which showed that there was increase. And levels of C3 and complement and other systems were there. There was decrease in metabolites like choline and lactate. Now at this time point, we did not get the answer why metabolites were reduced, choline and lactate, and why there was decrease in IgA, increase in IgA. And our findings were cited by NASA in their evidence report, because as I said, it mimics the NASA conditions, space conditions. So all these parameters, in addition to very many other parameters like hyperactivity, platelet activity we measured, inflammatory markers, complement activation, mucosal immunity. So because mucosal immunity was changed, there was change in the mucosal immunity, we thought that something has to do with the microbial changes, or there is some diversity or microbial dysbiosis is there. So why don't we look into that? So various publications came out of this work. So we started looking into the microbial dysbiosis. Is there any, is causing any change in the body system, that their mucosal immunity got changed? So we looked into the human microbiome. We all know that about 1 trillion cells are there. 60% of stool dry matter, which we processed, is a major mass. And there are various factors like age, nutrition, environmental factors which are affecting the microbial diversity, and the problems which eventually are led to our obesity, diabetes, heart disease, and other things. So we wanted to see who are they, which is a common question these days. Who are they? And whoever they are, what are they doing, and how they are doing it? What is the mechanism? What is it they are leading to? It's a treasure trove. So we put our project proposal in 34th expedition because we got some data in our 28th expedition. We proposed another expedition in 34th expedition, and protocol remained the same. We did not change anything so that there is a correlation between all the expeditions, collected the samples at the same time and similar samples, although subjects were different. Now, after collecting the samples, metagenomic methodology was followed, collecting samples and sequencing them, trying to find out who are they and what are they doing. And the metagenomic analysis workflow was followed as usual. Latest platforms were used, and quality control pipelines were followed. All statistical analysis were done. And then what we found that when we collected the saliva samples, what were the changes in the oral cavity? As you can see here, that there are changes in the bacterial composition, diversity was there, and you can see the pseudomonas was increased, which is not a good thing. It's an opportunistic bacteria, it causes secondary infections. And because people were confined in one place, they were sitting together, living together, so their droplets and other things could have caused infections. That was the major cause and of concern. And lactobacillus was also increased, which could have been because they were given loads of dry fruits and chocolates. And expedition members keep on eating chocolates, and that also puts on fat and other things. So these are the negative points which were occurring in the buccal cavity. Then we also found, functional potential. These bacteria which we found are changing. Are they contributing functionally? Are they changing the pathways or changing the metabolism of the body? Yes, we could find that there was glycan biosynthesis and metabolism were altered. Similarly, we conducted a study for gut microbiota. The stool samples which were collected were processed, and we conducted the 16S RNA sequencing. And here you can see that there was increase in E. coli. Now, the question which was raised in 28th expedition, that why choline and lactate were decreased, probably that was because of the E. coli, the answer which we got here. Because the tendency of E. coli is to break down the choline and other metabolites. So that could be one of the reasons which could correlate. And metabolites being signature of the cell stress, they are the snapshots which give us the idea about the whole body mechanisms. We also correlated with the functional analysis of these bacterial changes which occur, and that also showed that there is altered metabolism which is causing carbohydrate, lipid, amino acid, and energy metabolism changes there. So the changes in the metabolism, changes in the bacterial composition which is occurring is functionally correlated, and that shows changes. Now, we also tried to find out a few more metabolites if there are changes. We followed the proper protocol, and from NMR, we did the NMR, and we found that there are changes. We did the NMR spectra of serum and urine, and we found the functional correlation between the metabolic pathways, and there was change in the TCA cycle, histidine metabolism, pyruvate metabolism, and other major metabolic pathways will change. So that means that there is bacterial dysbiosis during expedition, and that has to be addressed. That has to be taken care of, and so that they could be the management of the Antarctic expedition could be better, and the expedition members who are facing problems like seasickness and other things which they are that could be managed. So these are the okay. Now, outcome of this study was discussed with NCR and Ministry of Earth Sciences. We showed them the data, and there were a series of discussions and meetings were held, and we showed them that how it can be, the management can be better, careful planning of expeditions, then limiting the supply of sweets and carbohydrates, for which has been done, and in forthcoming expeditions is also been done. All the expedition members are cursing us that why they have, because of our data, their consumption of, supply of chocolates has been reduced. Then optimal schedule of normalization circadian rhythm can be done, proper hygiene and circulation management can be done. So we put another project proposal that how we can manage. we had only one answer for that, that if we could use probiotic to maintain the body homeostasis, to maintain the gut homeostasis, which can help in regaining the gut structure. So we put up the project, and that project was through, and we started conducting studies using in 38th expedition, which is the story, a continuous story of 28th, 34th, 35th, and 30th expedition. We gave probiotic intervention to see that if that could help in maintaining the gut homeostasis. And in this project, again, the protocol remained the same, which we used for all last 3 expeditions, did not bring any change in that, and collected the samples in the similar way, so that it's a parallel study. Now, here we grouped the expedition members. When we suggested this project, it was said that Antarctica being the extreme environmental conditions are there, why don't we test it for the conditions which are mild? So we, at the same time, which we were conducting the experiment during Antarctic expedition, at the same time, on the same day, we were giving probiotics to our day candidates. They were used as controls because they were not under so much stress. So the grouping was done accordingly, and the protocol was followed. The day we started giving, right from the NCPR when they were at Goa, we started giving the probiotics every day, 1 sachet per day per person. Now, this is how it was given. Placebo group, one was the placebo group, where the same sachet was given, but without bacteria, and the probiotic which we selected after doing some better, and it was in a sachet, small sachet, and it was in a powder form, so that logistic problem also be resolved. This was the best way, and shelf life was for 2 years. So this is how the samples were collected at Trivandrum, Goa, before starting the probiotic and after completion of the ship journey. Now, what did we find? That the first thing which was of concern by Minister of Earth Sciences, that the expedition members suffer a lot of seasickness during ship journey, which happens during, even some have during bus journey also. So when we gave this probiotic, it was found that in 28th expedition, out of 30, there was 80% seasickness, in 34th, 66%, and after giving probiotic, there was only 10% seasickness. So that drastically reduced the seasickness of the expedition members. And that also gave us them good feeling, because they were not having any nausea, vomiting, or stomachache, or headache. So they were happy and generally having that good feeling, and they're doing all the work happily. So because the gut is the second brain of the body. So if that is fine, everything is fine. So that significantly reduced the seasickness. Along with this, when we did the taxonomic analysis, it was found that the more dispersion of negative correlation was, as you can see here, in placebo group than in the probiotic group. Green placebo, and probiotic was the red group. We also did the genus abundance plot, which also showed that there was, after completion of voyage, there was decrease in the PCB group, and Prevotella decreased in both the groups, Faecalibacterium increased, and that showed that yes, there is bacterial dysbiosis, which is changing the scenario or changing the body gut dysbiosis. E. coli was increased, as I said earlier also. Alpha diversity was higher in probiotic group than in placebo group. Beta diversity, well, there was more dispersion, distributed diffusely. And we can see that there was more abundance of bacteria in probiotic group than in placebo. That clearly indicates that gut homeostasis was well maintained after giving the probiotic. Now, again, we also did the function analysis of this study also, this expedition also, and we found that there was change in the pathways. In the placebo group, carbohydrate, amino acid, energy, fat, metabolism, everything was changed. Whereas in probiotic group, the metabolic alteration was regulated. It was not much. And mainly enzymes, the carbohydrate enzymes were abundant, and diversity analysis in placebo group. So that clearly shows that placebo group was affected badly than the probiotic groups. So that gave us a clear indication that if we give probiotic to our expedition members, that will help in managing them, their body status, as well as the expedition itself can be managed well. This work was published in scientific reports. Now, as I said earlier, Delhi we conducted the study, same study at the same time on the same day when the probiotic were given and when they were stopped. Same time was used for Delhi expedition. as it is said that some questions were raised that is it that if probiotic is taken in, without having any disease, not necessarily that we have disease and we take probiotic, what will it do? It doesn't do anything. Our daily data shows that there wasn't much difference. The metabolic pathways are well maintained and bacterial dysbiosis was not there. Everything was normal because we had kept both all the members were medically fit and no antibiotics taken, no disease was there. We had selected people, such people who had no disease or anything which was taken earlier. That shows that if we take probiotic normally also, it doesn't affect the body system. So with these parameters and with these study details, we proposed we gave the recommendations to Ministry of Earth Sciences. It was accepted as such. Probiotics doesn't do any harm. So this probiotic is being given in next expeditions for our Indian scientific expeditions from 38th onwards. Now, when we conducted this Antarctic study, we thought that our soldiers are deployed at high altitude at various altitudes. Why don't we try? Why don't we see what is happening in their gut dysbiosis? Because such studies have never been done because people, scientists don't get opportunity to get such samples. Our soldiers who are deployed at high altitude, being in defense, being in DRDO, we have this liberty and we get the opportunity of subjects who are deployed at high altitude. And they suffer from various problems. When they are at high altitude, the conditions of high altitude is similar to that of the Antarctica, in addition to hypoxia. We have hypoxia at high altitude. So that also puts another tremendous stress. And because of that, soldiers who are deployed have nausea, vomiting, diarrhea, peptic ulcer, inflammatory bowel disease, and gastrointestinal bleeding is there because they suffer from ulcer and colitis, which is a common problem. When they reach there, they have to take some medicines, some steroids they take, which causes peptic ulcer that damages their intestine, that damages the stomach wall, that leads to ulcers and bleeding, which is a matter of concern. And that was told by users. The user had asked us if we could find out something for our soldiers which can reduce the bleeding or the inflammatory bowel disease. So we conducted the studies and we got some regiment allotted to us. Sikh regiment was given to us where we collected samples at Bhatinda, and 213 samples were collected at 688 feet, 688 feet. All these soldiers went on to high altitude at 14,500 feet, remained there for 6 months. But as it is known that we cannot get the same number there. Some get posted out, some go on leave, some get retired. So the number got drastically reduced. But we could get 19 subjects where we could collect the samples, urine, saliva, and stool. And out of these 19 also, some of them went down to base camp because they had to go to further up, 19,000 feet. So we got the opportunity to collect these samples at various standpoints. The limitation of the study is number of samples, but the beauty is that it was a longitudinal study. They were the same subjects which we analyzed. So what did we find? That there was a phylum abundance. It is clear here. Yeah, genus abundance which we performed. As you can see here, H1 is the baseline, Bhatinda, where you can see the diversities, the bacterial composition, which was different from the H2. The H2 height changed. There was privotella, yellow colored, was abundantly present. When they came down to base camp, again, the composition changed and they went to 19,000 H4 height, again the privotella went up. But maybe by this time they got a little acclimatized, so scenario was a little different. As you can see, the bright blue band at the top of the third column, that is Fusobacterium. And this Fusobacterium is responsible for, excuse me. Nobody's here. Excuse me. Somebody here? Change. Yes. So ये चल ही नहीं रहा है. Okay, so there was microbial diversity here, and then we did whole genome sequencing and compared with 16S. It was same. That whole genome sequencing gave us the idea that, sorry, this is, yeah. Fusobacterium, which is responsible for thrombosis. That is another big major problem at high altitude. Many of our soldiers suffer from thrombosis, and it is not known, although it is treated, but lately now it has come to know that yes, there is, because of some bacterial infections, thrombosis is occurring. So that could be fusobacterium. So studies have to be done to find out that is it because of the fusobacterium or something else which is causing it. So it was compared to whole genome sequencing with 16S RNA, and we found the diversity there. It was there at H1, H2, H3 of all 4 heights. And we also compared the functional analysis H1 versus H2, H1 versus H3, H1 versus H4, and all the 4 heights did show changes in the functional aspects also, and species and strain level. We went down to the strain level, and we found that yes, there was changes in the metabolic pathways, which probably is causing the problem in the soldiers, leading to dysbiosis and gastric ulcers. And Prevotella copri 61740 was the major culprit, which was increased, and it is known that yes, Prevotella copri does cause problems. And this is what was found. As I said, the limitation of the study was very small size of the subjects, very small number. We suggested this data and put forth our recommendations to users, although it is not that we have to give the probiotics as such, because we will be getting a larger cohort. The trials will be initiated at high altitude, which is demanded by 14 and 33 Corps and Northern Command of the Indian Army. And studies, lab studies will be conducted for gut barrier injury using the same probiotic. So that is that probiotic helping in maintaining the gut dysbiosis at high altitude? We'll answer our question and help our soldiers to lead a better life at high altitude. So this is the team of people who have worked on it. And hopefully we will have some solution for our soldiers who are protecting us at the borders. Thank you.
Thank you, Dr. Lily Ganju. Very, very interesting topic. And you, when it started, I thought that the title is very different. But you know, you came to the point very quickly. And this is very interesting. Actually, this is a real translational research, that you do the research in the lab or in the field, and you come out, the outcome becomes a recommendation.
Yeah.
This is commendable. So if there are a couple of questions, we have time, couple of minutes.
I have one question, ma'am.
Sure.
Actually, when you are working with this probiotics intervention for looking at the dysbiosis normalization, so like whether the person who was consuming these probiotics, whether they're psychologically addicted to the same, or how long they should be, it is still the expedition is over, or they should continue with the...
No, sir. We gave for 60 days, sometimes 40 days, in other studies, 40, 60, that also helps. So we started from day 0, that is when they reached Goa. And from Goa, in Goa, they remained for 4 or 5 days. From Goa, they went to Cape Town by air. And from Cape Town, they boarded the ship. And the ship went to Antarctica, which is a very treacherous journey for 20 to 25 days, depending on the weather conditions. Sometimes is more than 30 days when they remain deep inside the ocean and ship starts rolling and pitching. So that psychological stress is also there. But by giving probiotic this time, in 30th expedition, they found that there wasn't that much fear because they were not vomiting, there was no headache, stomach ache was not there. So probably that helped in maintaining the homeostasis. And we stopped after the ship journey was stopped.
Okay.
Yeah, it should not be continued for a longer time. We can give a break.
How about the prebiotic intervention along with this probiotics?
Prebiotic, no, so we tried only probiotic.
Okay.
Only probiotic.
Any other question? So I will ask one.
Sir.
In your, I think, second set of experiments,
Sir.
once you found that chocolates are creating the menace, so you make recommendation that probiotics should be given and chocolates should be reduced. Okay.
That was accepted?
So, no. That was accepted.
Yeah.
Was the same experiment done that people are in Delhi? They are not being subjected to stress. They are giving chocolates and everything, whether they develop similar disease condition, and we should use human as a guinea pig. But these are simple experiments.
Yes, so that can be planned. Yeah, that can be planned.
Everything was, not everything. The major relation was that because they are eating chocolate. And the kind of food which is not normal food.
Yeah, yes. So similar food as they have, but of course not possible. Second, the chocolates in these 30th expeditions were reduced because we gave the recommendation 34th expedition. So of course it will not be same as Delhi, but yes, we can plan to have an experiment in Delhi by giving chocolates.
So with this, I thank you once again.
Thank you, sir.
Clap for good experiment. I think.
Thank you, ma'am, for your very informative presentation. We definitely follow your words, and we'll stop taking chocolates and we'll have probiotics.
Less gum problems.
Yeah, less problem. Yeah. I call dignitaries and the dais to felicitate Dr. Lily Ganju, ma'am. Thank you, ma'am. Thank you for. Thank you, ma'am. It's our pleasure having you here. We call upon next speaker, that is Professor Pamjit Singh Pansir. I request our chair and co-chair to introduce the next speaker.
Oh,
welcome, sir.
Yeah, thank you, Chair. Professor Pamjit S. Panesar is a professor in the Department of Food Engineering and Technology, and he has a vast experience and academic research of around 24 years, postdoc experience, and he is also worked in industry. So industrial experience also he has a lot. And he has been listed the world ranking of top 2% scientists. And also he is a INSA teachers. He has a INSA teachers award. He got it in the year of 2020. And he is a fellow award of food biotechnology, the British Research Society of India, and he has awarded as a better opportunity for young scientists areas of science and technology, and also young scientist fellowship, and awarded CSR. I mean, all the fellowship. He has a 150 research publication to his credit, 36 book chapters, 50 book reviews, and 110 plus papers been presented in the conferences. So with this, I invite him for giving his award lecture. Thank you.
Thank you, Dr. Harish, respected chairman, co-chairman, co-chair, and high dignitaries, the pillars of the society, Professor Kalu. He happens to be mentor of my mentor, and AFSTI fellows, Dr. Halimi is there, and Dr. Savanna, and other colleagues and previous speaker, Madam Lily Ganju. So it was an interesting talk, and Professor Verma is also there. So really it was a field work and actual service to the society. And I propose that probiotic chocolates can be the answer. And probiotic chocolates, people are working on that. That is there. And probiotic, prebiotic, synbiotic chocolates, the research is being going on. So anyway, coming to my just presentation, that is the valorization. In initial slides, maximum slide there towards students, because the senior students, senior faculty member, they all what is being presented over here. So that is why we call it research. So anyway, first slide to my institute where I belong to, that is in northern part of the India. and it is funded by Ministry of Higher Education, and we impart different engineering degrees and tech PhD. And under one roof, we have even KV school as well. That is, we can get entry for the first and get the PhD under the one roof. That is the beauty of our institute. So just coming to the story of this lecture, that is a food processing industry. Food is the essential part of our system. We have seen that. We have the lunch, we have the dinner, and we have the breakfast. And now we have processed foods. We have long list of processed foods there. And during processing, it is seen that waste is being generated, and approximately 40 to 50% waste is being generated. And even our kitchen is also a industry, and we can see how much waste is being generated in our kitchen. And if it is being processed some other way, then naturally it can be, the story can be different. And even there are different post-harvest practices there in the stores and grounds, and you know the picture, and stories are there in the newspaper as well. So anyway, so this is just overview of the food processing industry, where it stands, different areas where it is working. And under these sectors, different products are being made. That is, cereal is there, dairy is there, beverages, convenience foods, meat and poultry, fisheries. And then if I just see different unit operations, this is just summary of the unit operations which are being on different areas. And under the unit operations, different what we can call these waste are being generated, like solid grain, starch, gold, and different industries, different type of waste are there. And remaining, there are two more industries like poultry, meat, and then sugar industry. And you can see a long list. And dairy industry, you can see a weighing which is generated in a larger amounts, and you know the picture. And because CFTRI, you have the sister institute, and all work is going on there. So anyway, so this is just general characters of the waste, what type of waste is being generated in the food industry, that is organic in nature. And that is also biodegradable and contains good amount of the sugar. And so it can be easily isolated by the microorganisms. And so it can be tried for the different products by using the suitable microorganisms. So if I say what value-added products can be made, and from the byproducts, this is a summarized slide of the different products which can be made from the byproducts. And of course, as a microbiologist, because most of you are microbiology, you know that organism matters a lot, what type of organism you want to choose and what type of product you make to have. So just coming to the important, some of the important products where we had worked, and what is their importance and why we had chosen that. And this is just the organic acids, and skipping some of the slides. And if I say the organic acid, long list of organic acids, and these play a very important role when the food products are there. And naturally, they are the preservatives, the acidulants, pH regulators, many issues are there. And the acetic acid, you know, that is the cider vinegar, we know. So in this, we had worked on the lactic acid. Why we had used the lactic acid? And here I would like to point out just interesting things. That is, there are two isomers of lactic acid are there. One is the L-lactic acid and the D-lactic acid. And L-lactic acid is not easily assimilated by the humans. And chemically, we are not able to make the L plus lactic acid. Either it is racemic mixture or it is the D-lactic acid. So that is why we have chosen the fermentative pathway. And in this different, there is a long list of the microorganisms which can be used for the lactic acid production. And substrates, different substrates have been tried by different scientists. And of course, work has been done, but success has been obtained on the dairy industry, that is whey, using whey, and somewhat with the molasses, that is molasses have been used. So here another acid is citric acid. And citric acid has a very important role in industry, we know as a food scientist. And here also different substrates or byproducts have been tested for the production of citric acid. And much success is obtained with the using the molasses here. And these are the list of the organisms which have been tested on the various byproducts. And so mostly the Aspergillus niger has been tested when we are talking of the citric acid. Enzyme productions, that is the long list of the enzymes which can be used in the food industry. Their production is very sensitive when we're talking of, and in industry we are using the crude enzymes basically. This book is dedicated to that, where you can go through this book and all the enzymes, how we can produce, how we can use in different industry, that is there. So this is the list of the various enzymes which have been produced from the different substrates. Wheat bran has tested a lot when we're talking about the amylase. Pectinase is also produced from the apple pomace. And then we are talking of the lactase. Lactase, it is just a few minutes on that. That is a very important enzyme because lactose intolerance is a very big problem all over India. And even in the world, it is a big problem. And data shows that more than 60% person in India, they're suffering from lactose intolerance. So lactase can be the solution. In foreign country, they are using some tablets like lactase, and they are adding to a glass of milk, and you can do that. But this technology is not available in India. So people are working on that. Our laboratory has also worked a lot on this lactose hydrolyzed milk or lactose-free milk, and even the lactose hydrolyzed milk that has a different properties, and dairy products like ice creams and other things, they have different texture. When we're talking about the texture studies, they play a very important role when talking about even the sugar crystallization. That is low when we're talking of the lactose milk as compared to the whole milk. So that is the beauty of this enzyme. Then lipase, you know better. So here other area where byproducts have been tested, that is food additives basically. And when we're talking about the food additives, they play a very important role when we're talking of the flavor and texture of the product, because sometimes we say that it is a free flowing product, the texture is not proper. So the food additives, they play a very important role in that. And there is a long list of the food additives which have been produced from the byproducts when we're talking of it. This is summarized slide again. Then when we're talking about the polysaccharides, biopigments, bioflavors, biosurfactants, bacteriocins and vitamins, they have been tested. And even some amino acids. Vitamins, we are restricted only to vitamin B12 that is successfully produced, and then vitamin B6, that is the through the fermentation route. And when we're talking about the polysaccharides, and here just much work have been done on this spirulina, gallon, and even valine polysaccharide is being tested, and they have different properties when we're talking of the thickeners and the stabilizers. And then xanthan has been well tested. Xanthan from the apple pomace and molasses. A lot of work has been done when we talk of the xanthan. Then these are other studies where work have been carried. And here biopigments. Just here also I would like to speak. That is, colors are always attractive. That is a issue. And we always eat with the eyes. We call it. First we see the product, which is the color, and then we choose. That is whether we should eat or we should. Sometimes the color also discourages receptors to the food products. And even when we're talking of the synthetic colors, you can Google it. You have the in-hand mobiles. That is how many disadvantages are there. And even on the wholly, we say that is we should use the organic colors. And we are eating sometimes the dyes and other things. Even the limits are there, but sometimes we call it a biomagnification process. And ultimately, when you're exposed to daily some limits, ultimately they are dangerous. And even some skin allergies, sleepiness, that has been imparted with the help of the pigments. So biopigments, they can be the alternate. A lot of work is going on, and there are both the routes are there when we're talking about. Even fermentation route is there, and then green technologies are there nowadays. Ultrasonication, microwave extraction of the colors from the food byproducts. You can do that. You can just, we are talking about the sugar beet. You can extract the color from the sugar beet, and then you can add to the food products. You can extract the color from the carrot. So that is the also one system where people are working on that. And here in the fermentation, much work has been done on two microorganisms. First is the rhodiotrolla, that is glutinous, and which beta carotene has been produced by using this, and whey and potato waste liquid that has been used for the production of this. And even on the apple pomace, this rhodiotrolla has been tested, and on the whey also. So it produces a beautiful color, yellow color. And fermentation has the advantage that you can change the pH of the media, and end product can be different. So color can be different. So you can mix the two colors and to get the third color. The people who are having the art and craft hobby, they know that is we can produce a third color by mixing two colors. And even there is another organism, we call it a monaskis, and it produces a beautiful red color. And these two organisms have been tested a lot for the production of the biopigments, and they have successful. Only thing is instability with the light and temperature and long-term stability. There are issues. Now people are improving with the help of some of the other technologies like nano technologies, nanoemulsification, etc. So then bioflavors, you know, when you are having the fermented products that have typical flavor, you can capture that and then you can add to the other products. So same stories here. So here work has been done on the coffee husk, apple maize, soybean by using some microorganism. And then bacteriocins, we call them biopreservatives. The same story when we're talking about colors. Preservatives, some are the just salt is also active as preservative. What even then there are some chemical preservatives as well. And they have some side effects. So that is why people are after the bacteriocins. And in the bacteriocins, there are proteinaceous substances. They have been produced by using different microorganisms. Much work has done in the NDRA canal. So in the preservation of the cheese and other things. So here, that is different substrates have been tested. Even in our lab, we had tested different substrates, and they hasn't found to successful for producing the wide variety of the bacteriocins. And vitamin B12, you know, it is a well-known vitamin that has been produced from whey. So from the fermented root. So this is all about the basics, and then just a few slides what actual work we have done in the laboratory. Then we have the L+ lactic acid from the whey. We had developed technology. We were projected with the University of Birmingham, UK, and we had worked on that. And we know what it is, what it contains, and why we are again after the lactic acid. Now the single-use plastic has been banned from the 1st July. So what is the alternate? We want the biodegradable plastics. Now this is demand for the, when we're talking about L+ lactic acid, and we're talking about PLA, polylactic acid, and even the liquid crystal formation. That is very, expensive. Liquid crystal formation that is produced only by using the L+ lactic acid. So otherwise we cannot produce a liquid crystal as well. So PLA also use this chemical, and this is the demand for this. And then we use the different microorganisms, and we had procured some of the culture from abroad as well. We different matrices for the mobilization we have tested. And this is the outcome, that is we were able to convert the whey to a good amount of the lactic acid. And beauty was that is the system was stable up to the 16th cycle. 16th cycle, that is, this was the first report, that is we were able to reuse the immobilized cells up to 16 cycles. Otherwise, after 8 to 9 cycles, the cell degradation is there. The people who are familiar with the mobilization, some cell leakage problem is there. So we had developed some of the hardening steps and other things. So that is there, and this is just the same what I have told you, that is outcome of the study. And then we had a project with the PepsiCo, that we know that is the consumption of the Lay's, chips and other things, and how much waste is being generated in the industry. We know that. That contains a huge amount of the starch. So for that, we had also tested the L+ lactic acid. Study was also sponsored by the CSIR. So the organisms we had tested, and obviously we had optimized the process. And we had just tested both the bacteria as well as the Rhizopus orzi here, because the fungus has advantage because they are myelolytic in nature. So with the help of single step, we can produce the lactic acid, so we can reduce the time and other things when we're talking of the starch fermentation. The bacteria that was not successful, we were talking of the lactic acid production. Here we were able to recycle it up to the 8 cycles. So these are the some of the publication which we have published in different journals out of this work. So then another study we had worked on the lactulose, that is a prebiotic that we were talking of, the probiotic and prebiotics. And here we had used enzyme, we had produced enzyme in the lab. So I think within 5 minutes I will be able to compile it. So time is approaching. So here we had produced the beta lactose studies from whey because it has the double properties. One is the lactose hydrolysis, another is the trans-lactosylation activity. With the help of trans-lactosylation activity, we had used that property for the production of the lactulose. Lactulose, although it is available and it is produced through the chemical means, and it has some of the drawbacks. So we will discuss later on. Otherwise, we had developed a system to produce the lactulose from whey again. That is, it was successful. And again, we had used the mobilized cells, mobilized matrices, and this was the reusability of the mobilized cells for the production of lactulose from the whey. That is, there was almost, we were able to recycle up to 6 cycles of the this permeabilized yeast cells and enzymes. So then these are the related publication which we have worked related to the lactulose and prebiotics. And then another is a study we had carried on the galacto-oligosaccharides. These are also very good prebiotics, GOS and FOS. So here that was the study was sponsored by DBT. And again, we had used the mobilized beta-lactosidase from GOS. And we have just compared the pure lactose, then whey untreated and treated, because whey has also many impurities when we're talking of. Even some amount of fat is there, and that decreases efficiency of the cells. So we had compared all these things, and naturally that is much success on the pure lactose. What we were able to use the system on the treated whey as well. So that was the outcome. That is for the on the lactose, it was 14 cycles. On treated, it was 8 cycles, and untreated on the 6 cycles. So for the production of the GOS. So these are other such work which we are doing, that is on the bacteriocins we are talking of. And because in Punjab we have a kinnu, surplus kinnu, and kinnu byproducts, we are working on that for the production of extraction of biotic compounds by using various means like green technologies and then enzymatic ways, and then combination of these two. So that we are working on that. And these are other, that is the biopigments. I told you we are working on that. Even extraction of lutein. Lutein is also very expensive pigment. So we are working on that as well. So these are the other work which we are doing on, that is on the topical fruits like mango and banana, and then soybean meal. So that work is going on in the laboratory. So at the end, I would like to acknowledge the organizers who have just given me an opportunity to share the work with you. And it's my institute where I work and where I use the facilities, and my sponsors like DST and collaborators, University of Birmingham and DVT and CSCR for carrying out this work. So conclusion is apparent from the slides. And just one point, that is nowadays the biotechnological techniques such as mobilization, then genetic engineering, metabolic engineering, that can play a very important role. So that is there when we talk of the future where we can work on that. And this is just we have booked, and where for the student side, we have written a particular chapter on this lecture, that is from where I have prepared it, that is valorization of agro-industrial based. And this is a full book which we just published 15 days ago. That is for the valorization of the agro-industrial residues with the CRC. So you can go through. So this is just a take-home message, that is to avoid, to reduce the waste, that is judiciously use the 5 P's: petrol, power, paper, plastic, and potable water. And press the 5 R's: refuse, reduce, reuse, recycle, and be responsible. Although I have also taken from the net. So this is just for the students, so that we can be able to reduce waste where we can. So that would be contribution to the society as well as the nation. So thank you. Thank you very much.
Couple of two quick questions, if any, among audience. If there is none, then we thank the Professor Panesar sir for his award lecture. Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you for your valuable presentation. It was very informative. And I request all the dignitaries on the dais to felicitate Professor Paramjit Singh Pansir. Thank you, sir. Thank you very much. It's our pleasure having you here. I request Professor Satish to honor the dignitaries on the dais. Thank you, sir. Professor V.K. Chaudhary and Dr. K.V. Harish Prashant, CSIR, CFTRI, and rapporteur, and Dr. Jagdish S. from University of Mysore. With all your permission, we'll proceed with the next presentations. And now we will start AMI Young Scientist Award sessions. I request all the speakers to be ready. Yes, now we'll start AMI Young Scientist Award session. And it's my pleasure to introduce this session dignitaries. And in this session is chaired by Professor P.C. Abhilash. He is a professor at Banaras Hindu University, Varanasi. And he completed his PhD in Environmental Science in 2009, CSIR, National Botanical Research Institute, and the University of Lucknow, UP, India. And his expertise in the field of sustainability science, pesticide pollution, plant micropollutants, interaction under changing climate, rhizo-remediation of POPs, nanoparticles in soil system, restoration ecology, biomass, biofuel production from marginal and degraded lands, soil carbon flux, and microbial responses to climate change. He has published his findings in more than 45 research journals, and his total citation is 7,500 with a H index of 43. And his total impact factor of published papers is 160.193. And he is currently guiding the 4 PhD students. And he is a recipient of many awards like Travel Award from DST Sub to attend 10th Phytotech Conference at State University of New York, USA from October 2013. And also Travel Award from the Agrosciences USA to attend York Pesticides 2013 at New York University, and the Travel Award from the University of Oxford to attend the Planet Under Pressure, London. And is also recognized as visiting professorship, Sao Paulo State University, Sorocaba, Brazil, and visiting professor, School of Biological Sciences, University of Hong Kong, and Associate National Academy of Agriculture Sciences, NAS, New Delhi, and INSA Medal for Young Scientist, New Delhi, Yoshi Young Leaf Award from Yoshi Corporation, Japan, and Nazi Platinum Jubilee Young Scientist Award from the National Academy of Sciences, Allahabad, and elected a member in National Academy of Sciences, India, Allahabad, Young Scientist Award, and the Fellow International Society of Tropical Ecology, Varanasi, and affiliated member, International Union of Pune and Applied Chemistry, USA. Welcome you, sir. Welcome you for this session. And this session is co-chaired by Dr. Prakash Ji, scientist, IIRI, New Delhi. He got his postdoctoral degree from ICAR, New Delhi, and his expertise in the field of fungal pathology, magnoporte, host interaction, blast disease resistant, and his top 10 publications having NAS rating of 6 and above with the first and the corresponding authors only. And he is a recipient of many awards like Nanaji Deshmukh ICR Award 2021 and M.K. Patel Memorial Young Scientist Award 2020 and Best in Multidisciplinary Research Award in Palmillet 2015 to 2018 for New Delhi, and certification of recognition by the Indian Phytopathological Society for serving as a zonal counselor 2016, and recognition for multidisciplinary research involving basic strategies and applied sciences in different disciplines of agriculture. He has published his research work in many national and international journals with 5 patents. I welcome you for this session, sir. And this session rapporteur is Dr. Rashmi Kesi. She completed her doctoral degree from University of Mysore, and she is currently working as a faculty in University of Mysore, and she is having a 10 years of research experience. I personally welcome you, Rashmi.
Thank you so much.
Now we'll start with the sessions. I request chair and co-chair to introduce the first speaker.
Respected teachers, young scientists, scholars and students, a very good afternoon to one and all. On behalf of the organizing committee members and my co-chair, Dr. Prakashi IR New Delhi, and rapporteur, Dr. Rashmi Kesi. So I cordially invite all of you to this, one of the most awaited sections of this 3-day international event, AMI Young Scientist Award Series. So we are privileged to have with us 7 outstanding young scientists, those who have made exemplary contribution in frontier areas of microbiology, including the medical, industrial, agriculture, and other frontier aspects. So the first speaker in this section, Dr. Anugul Vaishnav. So I would like to call upon Dr. Anugul Vaishnav for his presentation. So he is currently working as an assistant professor at JLA University. He did his BSc, MSc and PhD in microbiology, biotechnology from Modi University of Science and Technology. His areas of research is molecular genetics and genomics, soil microbiology, below-ground interaction, and plant stress signaling. He has published within the short span of his research career, he has already published 30 research articles in international journals. He also a recipient of 2020 Young Researcher of the Award by Center for Education, Growth and Research, 2020 Young Scientist Award, Best Over Representation, and then he also received 2019 Young Scientist Award for the contribution in environmental protection. So with this brief introduction, so now I request Anugul to present his presentation.
Thank you, Abhilash sir. Hello and greetings to everyone. Myself is Dr. Anugul Vaishnav, and right now I am working as assistant professor in GLA University, Mathura. So here I am presenting some interesting finding of all over my PhD research work, that is about plant, soil and microbe interaction like how plant is communicate with the below ground communities for their benefits. So here I choose the topic plant communication with soil microbes for asking help under stress condition. So first of all, why we need to work on this, that there are some rationales on agriculture for coal for changes that are like increase food productivity by 50%. We have to increase our food productivity because like everyone you know about the population, the rising population. So we have to feed all of the population. So that's why we have to increase our food productivity. The second one is the farm productivity has been declined due to climate change, due to irregular uses of agrochemicals. And then climate change mitigation, yes, climate change also affect our soil health and the plant productivity. So we have to chase these rationales. That's why we are working on this field. Efficacy loss of agrochemicals, the efficacy is continuously losing. Negative impacts of food quality and human health, as we are using agrochemicals, so they create negative impact on food quality as well as human health. Then consumer demand for quality food. Now we are always demand for the organic food in the market. So that's why these are the reasons, that's why we choose this field. Here I use a figure where you can see that the overall land for the, overall land for the agriculture is around 149 million kilometers square. That is around 29%. But here you can see that the agriculture land where we grow our crop, that is only 23%. Out of 29%, there is only 23%. But still, we use most of the calorie and the protein from the plant-based food. So that is the thing, that we have the limited land and we have to increase our productivity in this limited land. That's why we are working on the sustainable agriculture area, like how we increase without the using of agrochemicals, without the using of breedings, how we increase our plant productivity in the limited land. So here I come on my topic, that how plant communicate with the below ground. So here some of the background about that, like soil microorganisms are responsible for the most nutrient transformation. These microbes are responsible for the biogeochemical cycle, phosphorus cycle, nitrogen cycle, carbon cycle. And here is the interesting thing, that the plant releases 20% of photosynthate in the below ground. That is the, if you imagine, that is the foolish thing. Like plant expenses about 80 ATP for one glucose, for making one glucose, but still they are releasing that 20% of photosynthate in the below ground. Why? Because these photosynthetes are responsible for the communication with the below ground, they are neighboring organisms that are maybe microorganisms, nematodes, and other neighboring plants. So these 20% photosynthetes are responsible for the communication with the below ground organism. And that is our main topic in which I worked on my PhD and my postdoc. Here you can see that if plant release volatile compound, then they may be the pesticide, they may be work as the pesticide, and then we can reduce the dependency on our agrochemical pesticide, chemical pesticide, if plant release such type of volatiles. Here on the right hand side, if plant release organic acids, then they will recruit the beneficial microbes toward the rhizosphere, and then they reduce the dependency on the agrochemical fertilizer, chemical fertilizer. So here, these microbes can be used as a biopesticide and the biofertilizer. Then this is the one of the hypotheses, like how plant recruit beneficial microbes toward their surrounding. So there is a one hypothesis, cry for help, like as we human, if we need any help, then we ask to other one. But in the plant case, they recruit some different type of, they release some different type of the compounds in the rhizosphere that are helpful for the recruiting beneficial microbes. Like here you can see, this plant release the different type of root exudates and they recruit the beneficial microbes. This is the mycorrhiza, or maybe these are the different PGPR. And in reverse, these microbes help for plant to induce resistance against biotic stress. Likewise, here you can see these microbes also produce the pathogen inhibitory compound that directly reduce the growth of the pathogen in the below ground region of plant. So I worked on this hypothesis. I try to prove this hypothesis in my PhD work. This is the part of my PhD work. We extract some soybean root metabolites in the presence of salt stress and under the normal condition, and we check the chemotaxis assay of these metabolites towards a beneficial bacteria that is name is Pseudomonas simei AU, and that is submitted in the MTCC culture collection. So here you can see that when we use the root exudate that release in the presence of salt stress, these root exudate recruit more number of Pseudomonas simei cell in the rhizosphere region. And when we check when we analyze through the LCMS the compound name, these green highlighted compounds were newly expressed in the presence of stress condition. These compounds were newly expressed in the presence of salt stress condition in soybean root exudate. So here you can see that it is true that the soybean plant release different type of root exudate in the presence of salt stress, and maybe these root exudates are responsible for recruiting more number of the bacterial colonies towards the rhizosphere region. And these bacteria is helpful for relieving abiotic stress, for relieving salt stress in plant. You can see in the next slide. And in response to root exudate, we also analyze our bacterial volatile profiling, like in reverse what bacteria is doing in response to root exudate. So here you can see that these yellow highlighted compounds were newly expressed in the presence of salt stress root exudates, these compounds. And if you Google these compounds, you can find that hexanol, quinoline and tridecane, they are responsible for the plant growth promotion. And these nitroglycol and benzothiazole, they are responsible for the pathogen suppression. So these compounds we found in our bacterial volatile profiling. And when we perform our experiment with the plant growth, so this was our first time in the 2015. So I just used this setup. What I did, I just put our bacterial culture on the lid of the magenta box. Here you can see, this is the upper side. And I grow my plants in the bottom. So there is no physical contact, because here I want to check how bacterial volatiles affect the plant growth. So this was our first experiment, and we found that the root hairs were increased in the presence of bacterial volatiles. And because the leaf part was most exposed in the presence of bacterial volatile, because this is the upper part. So we check some protein profiling in the leaf part, and we check some gene expression in the leaf part, and we found that the rubisco, gamma glutamyl hydrolase and vegetative storage protein were expressed, highly expressed in the presence of bacterial volatiles. And these are some salt stress responsive gene. So their regulation is also increased, like peroxidase, catalase. This is for proline synthesis, this is for vegetative storage, and this is for sodium potassium transporter. So here we can see that the bacteria that was recruited by the soybean root exudates, that bacteria help for plant growth promotion. So our hypothesis is proved here. Then we move on the biotic stress, because first is the abiotic stress, now we move on the biotic stress. Here we use different bacteria, Bacillus species SJ5, that is also submitted in the MCC. Now the culture collection name is changed in IAMR in the Pune. And we found that in the presence of fungal phytopathogen, that is Fusarium oxysporum, and this is the Rhizoctonia solani. So in the presence of fungal phytopathogen, our bacteria release different type of the volatile compounds. So here the green ones are the different, that is released in the presence of the fungal pathogen. This is for Fusarium oxysporum, this is for Rhizoctonia solani. So here again we can prove that in the presence of microorganism interaction, like our bacteria present in the rhizosphere, so they can interact with the other fungal pathogen also. So in the presence of fungal pathogen, they also release different type of compounds that may be the responsible for the suppression and the growth promotion of plant. Then in the 2021, we use a cool setup for the volatile interaction. Here you can see that we use two different compartments. This is our port compartment, this is our microbe compartment. Here we inoculate our trichoderma, and that is connected by the pipe only in the below ground region. So here the volatile will be released in the below ground region, and that will impact on the root part of our plant. So we found these findings in this experiment, like this, the trichoderma producing volatile that suppress the Escherichium rollsi, that is one of the phytopathogen. And this volatiles also increase the lateral root formation, and also increase the defense against the S. rollsi. So here you can see the result. This is the control S. rollsi, and if we inoculate our trichoderma, this is the BHU2 that we finally choose, that reduce the growth of the fungal pathogen, and that is bipartite petrified. That means there is no physical contact between two microorganisms. And here you can see the seed germination effect, and here you can see the degradation of the hyphae of S. rollsi in the presence of our trichoderma volatiles. This is the control, and in the bottom, this is in the presence of volatile, so you can see the degradation. And because the root part was most exposed in the presence of our volatile, that's why here you can see the lateral root formation was increased when we inoculate our trichoderma in the second compartment and the volatile transfer with the pipe in the root compartment. So this was our newly study. And we also checked in the presence of synthetic media, PDA, and the soil media. Like if we change the media composition, microbes change their volatile profiling. So here you can see in the presence of soil, only around 30 compounds released, and in the presence of PDA, that is complex medium, synthetic medium, so the compounds were around 46, and only 9 compounds are common, but the most of the compound shows antifungal property. If you Google these compounds, you can find these compounds are showing antifungal property. So we use PDA and soil as well, both in our second compartment where we inoculate our trichoderma, and we check the volatile profiling through GCMS analysis. Then this is my latest study. I was selected for a Swiss Excellence postdoctoral program. So this work was done in the University of Zurich. Here we also try to identify how mycorrhiza transmit stress signal to the soil microbes. So here you can see, like we select such type of setup where we add our 2 petty plates to the pot, where one petty plate was covered by only 0.5 mm mesh and one petty plate was covered by 20 micrometer mesh. 20 micrometer mesh means only hyphae can transfer this side, and 0.5 millimeter mesh means root and hyphae both can transfer. So here our aim was that how mycorrhiza transfer stress signal by hyphae and by root as well as the mycorrhizosphere. Like we can say this part is the mycorrhizosphere, means where hyphae and the root are attached, and this is the hyphosphere where only hyphae is attached. So we design 4 treatments. This is the control, this is the pathogen infected. We infect our plant from the foliar part, and this is our AMF inoculation, and this is AMF plus pathogen. So we found that the inoculation increase the, like here you can see this is AMF plus pathogen. It increase the total biomass as compared to the only pathogen infection. And here you can see the total leaf area also in comparison to pathogen. And here the stem diameter, because when we talk about the pathogen was Fusarium verticilloides. So if we talk about the infection, there are chances to reduce the stem diameter. But here you can see that in our AMF inoculation, mycorrhiza inoculation, the stem diameter was increased in the presence of pathogen. And when we check the bacterial diversity for both compartment, for root compartment where root and hyphae were transferred, and for hyphae compartment where only hyphae can transferred, we can see that there are changes in bacterial community in the presence of healthy and pathogen plant. You can see here. So here I mentioned that the most significant changes takes under AMF plus pathogen. Treatment belongs to Microciliaceae, Pyraliaceae, and these other families. Here we can conclude that yes, mycorrhiza transmits some stress signal when we infect our plant with the fungal pathogen. And this stress signal is also transferred by the hyphae, that is not attached by the root, and also transferred by the root in the both compartment. So this work was recently done. And finally, my key consideration is if we use approach of microbe-based approach in the agriculture, then their application can deliver different type of benefits, and we can cover different type of sustainable developmental goal. Here I mentioned like SDG 2, we can chase by the increasing farm productivity. If we use microbe-based approaches, then the farm productivity will definitely increase. Then SDG 1 by the farm profitability, SDG 3, 6, 13 and 15 by nutrient quality and reducing the agrochemicals uses. And if we produce more bioformulation, then there definitely by creating new industry for the production of microbial products and formulation. So we can chase these sustainable developmental goal. And finally, yes, sir, I'm just compiling. So these are the achievements. I published more than 15 publications, including research article and book chapters. Recently, I granted one patent from the Indian Patent Agency. The number is this one, and this is based on the controlling fungal pathogen. And 9 different patents are still under the examination that are also based on the microbial based formulation. And I edited 8 books published in the Springer, Elsewhere, and CRC Press. These are some technology that we developed. This is the biopot, that is as an alternative for biodegradable nursery growing pot. Like in nursery, we use only the polythene, black polythene for holding the soil. But here we use this pot. You can directly put this pot in the soil, and this is also helpful for the plant growth promotion and disease suppression. Here we use encapsulated bioprime seed, and we design a nano formulation for biofungicides. These are my books that published in different publisher. And finally, I acknowledge my mentor, Professor H.B. Singh. He retired from the BHU. Professor Marcel van der Heijden, he is in the University of Zurich. Professor B.K. Sharma, he is working as a professor in BHU. Anil K. Saxena, he is retired from the ICR NBIM. And my PhD supervisor, Professor D.K. Chaudhary, is working in the MIT Noida. Dr. Franz Bender, he is also working in the University of Zurich. Professor Kandi Prakash Sharma, he is working as in the Central Institute of Haryana. And my colleagues and students, thank you.
Thank you, Anugul, for your wonderful presentation. The crosstalk between plants and microbe, and how the plants are asking help from microbe through signaling or infochemicals, and most importantly, how this partnership can be harnessed for achieving UN Sustainable Development Goal. Now the presentation is open for discussion. We can quickly take 1 or 2 questions from the audience. Any questions?
I think the back side there is one.
Yes, please go ahead.
When you compare it with the agrochemicals which is used, what is the relation in your study? What is the risk, percentage of the risk of the pathological disease?
Can I ask your name?
I'm Tahrin from CSR CFPR.
Thank you, Tahrin. If we use our microbial formulation, and that is depend on the different type of microbes. So if we talk about the trichoderma, if you not use any agrochemicals, trichoderma can reduce up 60% of disease incidence. And if we use half portion of the agrochemicals and half portion of the trichoderma, then you definitely chase the more than 80% disease incidence that can reduce more than 80%. But it depends on the microbes that you are using. Am I clear?
Okay, thank you so much. Once again, we thank you for your excellent presentation.
Thank you
so much.
Thank you, Dr. Ankur Vaishnav. I request dignitaries to give a token of memento to Dr. Ankur Vaishnav. Thank you, Dr. Ankur Vaishnav. The next speaker is Dr. Gaurav Sharma. I request Dr. Gaurav Sharma.
Welcome, Dr. Gaurav Sharma, and congratulations. I would like to briefly introduce about Dr. Gaurav Sharma. His current designation is he's a DST Inspired Faculty Fellow, especially on microbial genomics. He did his PhD and postdoc both on microbial genomics. He had a PhD from Chandigarh, CSR, in Tech Chandigarh, and postdoc from Department of Microbiology and Molecular Genetics, University of California, Davis, USA. Then his research interest, if you see, I said mostly on microbial genomics, and he was fascinated in microbiome also, metagenomics, etc., and all computational biology also. He received award in faculty fellowship from the Department of Science DST from 2019 to 24. He has been recognized for his PhD work. He has sequenced 15 novel myxobacterial strains and did a comparative genomics work. And later he worked with Professor Mitchell Singer, University of California. With this brief introduction, I would like to welcome Dr. Gaurav Sharma for his presentation. All the best.
Thank you. So first of all, I would like to thank AMI for giving me this opportunity to present my work. And so right now I'm working as a DST Inspired faculty at IIB Bangalore. This is my group right now. And my lab is supported by DST, and we also have one funding from ICMR. So today I will be talking about my PhD work and my postdoc work and current work also, which I am doing at IBAB. And broadly, our model organism is Myxobacteria. So till now, most of the talks has been focused on experimental biology and a little bit of computational biology, but this work is completely computational biology. So I will be talking about understanding the physiology and evolution of these soil-dwelling Myxobacteria using comparative genomics. I have been already introduced, so I will skip this. So the focus area of my lab is fourfold. So we are highly interested in microbial genomics and evolution. I have been working on Myxobacteria from last 10 years. And recently we have also started working on Vibrio organisms to understand their physiology. And in the last 2.5 years, we have also worked on SARS-CoV genomics, and we published several papers on that. Simultaneously, my DST Inspire project is more focused on medicinal plant genomics. So we are trying to understand that how medicinal plants and microbes are interacting with each other. And is there a way out where microbes are helping in synthesizing those medicinal compounds in those medicinal plants? But again, this project, we are just starting. And we are also, we are also involved in making prediction servers. So where, biologists who doesn't have too much computational knowledge, they can directly use these softwares for understanding of their organism's genome. And we are also collaborating a lot. So just coming to the main topic. So the first question comes that, why we are interested in Myxobacteria. So Myxobacteria is really one group of fascinating bacteria. And I'm sure by the end of these 2 slides, you will be also fascinated by these organisms. So the first property which they have is unique taxonomy. So if you see, they belong to Delta Proteobacteria. I'm not sure if you are able to see the... Okay, so they belong to class Delta Proteobacteria, and most of the Delta Proteobacteria are anaerobic nature. But Myxobacteria, they are all these organisms which I have listed here, they are aerobic. So basically, these are the only aerobic group of organisms within Delta Proteobacteria, which make them very interesting. Second is their complex physiology. So they depict social behavior. They are they live in swarms, all together. They do these adventurous and social gliding motility. So if they are in a swarm, they do social motility. If they are alone, they will do adventurous motility. Based on their social behavior, they also make fruiting bodies. They secrete large number of secondary metabolites. They degrade so many biomolecules. And one very interesting point for at least a computational biologist is that they have a gigantic genome. So if you think about E. coli or Bacillus, these group of bacteria are 3 MB, 4 MB, maybe 5 MB. But these group of organisms are 9 to 16 MB genome, which is almost 4 times as compared to Bacillus or E. coli. So the question which we had in our mind is that, what is the requirement of having this large genome in these organisms? And if you see this graph, the red ones are all the Myxobacteria. So if you see, it means that all the largest genomes belong to Myxobacteria. And you will find during my PhD, we also sequenced the largest bacterial genome known so far, which belongs to Myxobacteria. One of the major, relevance of Myxobacteria to human being is based on their secondary metabolites. So this is a recent paper in 2021 where they talk about, the 20 topmost organisms which are secreting secondary metabolites. And if you see, it has streptomyces, which has been highly studied in terms of secondary metabolites. But if you see these arrow, marked organism, all of these are older Myxococales, and they are one of the largest producer of secondary metabolites, and especially hybrids of NRPS and PKS type of secondary metabolite, which is very novel in the bacterial kingdom. So as I said, that this is a less explored territory with huge potential. Means right now, at least the work which I will be talking about is more focused on understanding their physiology, but in the future we are planning to work on their secondary metabolites. So just to give you a quick introduction that what I will be talking about here, that so we, in the starting of 2011, when I started my PhD at Tech Chandigarh, I started with procuring several of these strains from DSMZ. We cultured them in our laboratory, and we did the genome sequencing, and we further later tried to understand a lot of physiological properties of these organisms using comparative genomics, and I will quickly discuss several of these results here in next slides. So After receiving these cultures from DSMZ, we cultured them, and now we are also trying to isolate them from their natural environment, either as a pure culture or part of metagenome. Later on, after culturing them, we try to go for genome sequencing, and we have used different type of genome sequencing strategies like PacBio, Illumina, Roche to get all these genome sequences. So as you can see, all of these pink highlighted ones have been sequenced by me during these last several years. And just to quickly show you here in the genome size plotter, if you are able to see that all of them are almost like 9 MB or 10 MB, and this like one of the, like the second one, if you see from the bottom, it is like almost 16 MB, which is like the largest genome in this bacterial kingdom, which has been sequenced by me. Now, so the first thing which when we procure any strain from any culture collection, we always try to identify that either it is the same strain or not. But what we found in one of our studies that we procured one strain as the Chondromyces apiculatus, but based on our genomic studies, we found that it doesn't belong to that particular strain, and we were able to characterize it in a novel strain which is known as like Myxococcus hensipus, and we did it using like 16S phylogeny, housekeeping gene phylogeny, genome distance, and all-to-all protein comparison. And we published this work in PLOS ONE. So as you can see here in the table, that Myxococcus hensipus is showing the highest similarity with fulvus with just 32.7 DDH value. To belong to the same species, this value should be like around 70. So we propose that this organism will be like a novel species within this genus. We were also interested in understanding their biomolecule degradation. So we have studied like all of these genomes to understand that how many enzymes they are secreting, and again based on computational analysis, we identified that they have a very large plethora of enzymes, and we have also worked on several of these categories like chitinases and amylases, and what we have found that most of these enzymes have been procured from different bacterial groups into this particular group of organisms, suggesting that when they are living in the soil environment, they are trying to adapt whatever is useful for their life cycle. They just try to grab it from other group of organisms, and we have published these two works in Genome Biology and Evolution. One of the most interesting thing which we have done is to try to understand their signal transduction potential. So just to give you like very quick introduction, that usually bacteria try to respond to the environmental stimuli using either transcription regulators or two-component systems, or there is another high complex like a complex version of two-component system, which is chemosensory system. So in transcription regulator, the signal, like which is these red dots, they are directly received by a ligand binding domain, and they either activate or repress the transcription. But in two-component system, the sensor kinase receives the signal, and then it transfers the signal to response regulator, and then it activates or repress the transcription. But in chemosensory system, the signal is received by these chemoreceptors, and these chemoreceptors transfer the signal to KA, which is a histidine kinase, and then KA to KY, which is a response regulator. And then these KY, which is response regulator, they can be involved in like so many alternative cellular functions like flagellar motility or type 4 pili motility, or maybe like biofilm formation or flagellar biosynthesis. So what we did, we tried to identify all these components of chemosensory systems within our myxobacterial genomes, and we found that again, just to compare it, that E. coli has only one chemosensory system which is involved in motility. That's it. But these group of organisms, they have like up to 12 chemosensory systems within them, and they are involved in so many different functions, as I mentioned earlier. Similarly, so in this way, we were able to identify 6 new chemosensory system classes within these group of organisms, and based on our synteny analysis and computational analysis, we were also able to predict the function of 2 of the chemosensory systems, and we published this work in Journal of Bacteriology along with their cover page for the journal. So as you can see, like this is a standard, this is a phylogeny of Ka proteins which are involved in chemosensory systems, and in this way, all the chemosensory systems are one monophyletic clade, and which are different in each other. Okay, and in this way, we have identified 6 novel chemosensory systems. So if you quickly have a look at this table, which is a representative of that which chemosynthesis system is conserved across all, you will find that key 8, this system is the only one which is conserved across all, and others are relatively present in different group of organisms. So later on during my postdoc, I found that this particular chemosynthesis system CC5 is present alone in this, it's not present in any other group of myxobacteria. And when we found that this particular methyl chemoreceptor protein, it has a special domain known as PAS domain, PAS domain. And this PAS domain is involved in sensing oxygen or redox potential and light. So basically, this is more of an energy sensor. And we suggested that this particular system will be involved in energy taxes within this this group of organism. And when we performed like its homology studies, we found that this particular chemosensory system is only present in actinobacteria and cyanobacteria and not anywhere else. So based on that, we suggested that this particular system might have been acquired from actinobacteria and cyanobacteria, which might be living like nearby via horizontal gene transfer. And we published this work in BMC Genomics in 2018. So as I said, like the myxobacteria perform two types of motility. One is adventurous motility and other is group motility or social motility. So social motility, they do use typhor pili. So these typhor pili, they stretch the typhor pili and then they pull themselves in the direction. Whereas for adventurous motility, they take the help of slime and these focal adhesion points. So we try to explore a little bit into these directions. So as I said, the typhor pili are involved in these like social motility. So we try to study all the genes which are involved in these typhor pili, and we found that they are making two types of groups here. So one is like, as you can see, the upper panel, and the second panel is like, the scattered genes. So, and when we compared this organization within Delta Proteobacteria, we found that the group kind of organization is present in these two group of organisms like bdellovibranales and disulfomonadales, and the scattered kind of distribution is predominant in other group of organisms like bradymonadales, disulfuroales, or disulfobacteriales. And so in this way, there has been like a bidirectional evolution from Delta Proteobacteria for the typhor pili system. And we published this work in Frontiers in Microbiology. Simultaneously, we are also interested in...
You have 2 more minutes.
Okay, okay. So I will try to skip a few slides in that way. Okay, but... so we also try to study their transcriptome to understand that, how many proteins are involved in different time frame. So so myxobacteria usually do this 72-hour life cycle. So they have vegetative phase and low nutrient phase. So we try to, we did some transcriptomic studies and identified that, like different proteins which are involved in different time frame for their development. And simultaneously, we also found that one particular group of cells, which are peripheral rod cells, which support the myxospores, which are germinating within the fruiting bodies. So these cells are different type of cells, and they are not normal vegetative cells. So we again confirmed this using transcriptome studies, and we published this work in Genomics. And I will skip these two. Maybe this slide I will skip. So we are also interested in their polysaccharide synthesis to understand that, how they are making biofilms, how they are communicating with each other. And for that, we identified these different mechanisms for polysaccharide synthesis, like WZX, WZY kind of pathways. And later on, we also identified that, we identified that in myxobacteria, like if you see these 3 proteins, like EPS, MASC, and BPS, they don't have this particular domain which is involved in polysaccharide secretion out of the cell. So based on our genomic studies, we suggested that there is this particular beta barrel protein which is present nearby, and this particular protein helps in secretion of the protein. And this work has been recently accepted in Microbiology Spectrum, an ASM journal. So concluding my talk, that we have been working on these different dimensions to understand myxobacterial physiology and how they might have evolved, how they might have procured this much DNA for their development, for their life cycle. And just to quickly mention that we have also worked on these SARS-CoV-2 diversity and evolution, and some of the work which we have published in PRJ, Journal of Medical Biology, Nature Biotechnology, and Frontiers in Public Health. And with that, I would just like to acknowledge my lab members who have been working with me from last 3 years, and my mentors and collaborators, especially Professor Mitch Singer at UC Davis, Dr. Sri Krishna at CSR Imtech, and several collaborators with whom we have been working, and funding agencies, DST, Inspire, ICMR, and yeah. Thank you. Thank you for listening.
Yeah, thank you very much, and congratulations. A great piece of work, especially on myxobacteria and their evolutionary systems through genomics as well as transcriptomics approach. So I'd like to, from the audience, if any queries, 1 or 2 questions can be. Yes. Yeah, can anyone please pass the mic?
Hello, sir.
Yeah.
Generally, bacteria like Bacillus or E. coli have a GC content of 43 or 50%. But this bacteria, as you mentioned, it has 70% of GC content. Does it have any significance on the evolutionary relationship? That's why it is very far related or anything like that? The more GC content constituting to more number of amino acids in that way?
So there have been like some research where people have tried to understand that does GC content has any correlation with life cycle or correlation with any other factor, but they are not really able to correlate it. Okay, so a few points which people have recently identified is that sometimes if like some organism has high GC content, maybe they will have like slow growth in nature. So, and at least it is proportional for myxobacteria, because myxobacteria are very slow growing organism. So yeah, but there are no correlation as such, because there are streptomyces or actinobacteria, which are also high GC content organism, but they are kind of high growing like organism. Yeah. Yes, yes, definitely. But still we feel so much advantages in this particular. So the good point about this particular group of organism is that it's not pathogenic. It just living freely in the soil. So it's not harmful for anyone. And the advantage may be that it has secreting so many secondary metabolites. So right now it is not like they have not been explored too much. Yeah.
Thank you. Thank you very much.
Okay. Thank you.
Thank you, Dr. Gaurav Sharma. I request dignitary to give a memento to Dr. Gaurav Sharma. Thank you, sir. The next presenter is Dr. Satyanarayan Patel. Dr. Satyanarayan Patel.
Okay, thank you. Moving on to the next speaker, Dr. Satyanarayan Patel. He's working as a scientist there in Center of Innovative and Applied Bioprocessing. He's already an accomplished young scientist, published almost 10 publications. He has also received prestigious Young Scientist Award like Gandhian Young Technological Innovation Award, Anna University Center of Excellence Best Researcher Award. And he also selected for a competitive B4 synthetic biology workshop. And then he also having 4 patents in his credit, and he already qualified all fellowship. I mean, he already got GATE, CSAR fellowship and all that. With this brief introductions, now I request Dr. Ranjit Satyanarayan Patel to present his finding, please.
Very good afternoon to all of you. I am Satyanarayan Patel and recently completed my PhD from CIIB Mohali.
So the today's talk title is a novel D-allulose 3-epimerase variant for the production of a functional sugar of anti-diabetic and anti-obesity potential D-allulose. So as we know that the worldwide prevalence of obesity and diabetes is increasing day by day, and according to a report by the World Health Organization, more than a quarter of world population is obese. Similarly, according to International Diabetic Federation, forecast predicted that the diabetic individual may increase from 463 million to 700 million by 2014-15. And n number of literature suggested that an excessive consumption of caloric sugar are responsible for these obesity and diabetic. So if we can replace these caloric sugar with the rare sugar, so we can reduce the diabetic and obesity incident all over the world. So what are the rare sugar? Basically, rare sugar are natural sugar, and it's a reduced caloric sugar. So one of the very demanding rare sugar nowadays, that is the D-allulose or D-cyclose. So D-allulose, also known as a D-cyclose, is a C3 epimer of D-fructose. It's an ultra low and low calorie monosaccharide, and showing a number of physiological functions such as a low glycemic, blood glucose suppressive, decreasing adiposity, anti-dyslipidemic, neuroprotective effect, and antioxidant property. Apart from that, it also enhances the storage activity and gives a better mouth feel. So there is an enzyme that is called D-allulose 3-epimerase that converted D-fructose that convert D-fructose substrate into a allulose in the presence of this enzyme. And the daily intake level of this enzyme was 0.9 gram per kg, and generally recognized as by FDA and in India FSSAI. But the major problem with this enzyme is the poor thermal stability and low yield of enzyme. So basis on this problem, we started working on this enzyme. So the N-terminal fusion of SMT3 into agrobacterium tumefacient D-allulose 3-epimerase. So with the first problem, we want to increase the thermal stability of enzyme. So basis on literature, we have fused SMT3 at the N-terminal tag of the native enzyme, and then we characterized, and then we found that the modified enzyme showed 15-degree higher optimum temperature, that is 65 degree compared to wild type. Later we checked their thermostability, and we found that the wild type enzyme showed 70 minutes at 50 degree, whereas the modified enzyme showed 943 minute half-life at 50 degree. That is a 14-fold higher than the wild type. So this technology was transferred to one of the Indian-based company. Later, we have immobilized this modified immobilized enzyme into a magnetic nanoparticle by covalent coupling, and then characterized through n number of techniques such as SEM, TEM, etc. Then later we checked their half-life. So we found that at 50, 55, and 65 degree, the immobilized enzyme half-life was again increased 4 to 5 fold compared to free modified D-allulose 3-epimerase. And later we checked their recyclability. So we found that this immobilized enzyme after 10 cycle only reduced 10% activity. So that means we can produce at least 10 cycle with the same enzyme. So it will helpful for the economical production of this product, that is a D-allulose. This technology also transferred to one of the Indian-based company in 2017. As in my lab, many students are working in a metagenomic database. So I thought, why not I can use these database to discover a novel type of D-allulose 3-epimerase. So I was used one of the data from hot spring and perform the blast through the known D-allulose 3-epimerase, and there is a n number of bioinformatics analysis. I shortlisted more than 50 genes, and one of these gene that is working as the D-allulose 3-epimerase So later we have characterized that gene and expressed successfully in E. coli and Bacillus. And that gene, the novel gene was active in the pH range of 6 to 10 with the optima of 7, and the optimum temperature was 75 to 80 degrees. When we check its half-life, we found that this enzyme active at 9,900 minutes at 60 degrees, whereas at 70 degrees, 3,003 minutes. When we check, we compare this data with the non-Cellulose epimerase, so we found that this enzyme was highly stable. Yeah, we can compare to till now reported any Cellulose epimerase. Its catalytic efficiency and turnover number was also good. Further, as we know that there is a n number of juice are available with the caloric sugar, so we thought why not we can convert presented the fructose into allulose, and we successfully converted fructose into allulose in these juices. As D-allulose is a sugar that is a food grade thing, so we thought why not we can search D-allulose epimerase from food grade strain. So I was mined NCBI Bacillus species database. From that I found one gene, and that gene was active at a pH 7 to 8 with the optima of 55 degrees. And this is the HPLC profile where you can see it converted the fructose into D-allulose in the presence of this enzyme. Here you can see this enzyme active at 36,000 minutes at 50 degrees. So again, this is showing highest thermal stability at 50 degrees compared to any known Cellulose epimerase. Here you can see the kinetic parameter. So this shows a good turnover, that is a 367 seconds. So this is the second highest turnover number reported by any Cellulose epimerase throughout the world. As many companies came to our lab for the commercialization of this product, through discussion we found that there is a major problem with this enzyme. First is the stability, another is the low protein yield. So yeah, we have previously two protein, but there is a good property, but the yield is very low. So again we started searching and we found that one gene. You can see this gene was active from pH 4 to 95 degrees with more than 80% activity, with more than 80% activity with the optima of 90 degrees. So this enzyme fulfill the first criteria, that is it's active at all the tested temperature. And then later we check their protein yield. So we found that this enzyme showed 180 milligram per liter in sick flask culture. So this protein fulfill all these the questionable two things, that is their thermal stability thing and the yield. So the patent manuscript under preparation. And another thing is for the economical way production of D-allulose. We have tried to extracellular secretion of D-allulose epimerase. So we have cloned this enzyme into a PST43 vector with the amylase signal sequence and successfully transferred in a Bacillus subtilis and also expressed in a medium form. And right now we are working with the IMTech scientists to standardize and secrete at fermentation level to demonstrate the industry. As D-fructose is a little bit costly substrate, we thought why not we can produce the D-allulose from using the cheaper substrate glucose. So for that we need two kind of enzyme. One is the xylose isomerase, another is the D-allulose epimerase. So xylose isomerase can convert D-glucose into D-fructose, and later D-allulose epimerase will convert D-fructose to D-allulose. So for that we have again I need this enzyme. So again I mined metagenomic database and found one of the best efficient xylose isomerase from that database and characterized. And further after characterization I just standardize the doses ratio of the enzyme, which of the enzyme ratio is good for the maximum level production of D-allulose from D-glucose. We found that 3:4 is the good ratio for the maximum level production of D-allulose. And here we can see when we applying both the enzyme xylose isomerase and D-allulose epimerase, the allulose is converted into fructose, and later on it converted into D-allulose with the 18.2%. So this is again highest conversion reported by any group in the world. So in future prospectus, high level secretion of enzyme by fermentation approach and genome engineering of the Bacillus subtilis for high level production of D-allulose. And these are my publication. Some of the Indian patent granted, and a few of the have been already filled, and 4 patent has been licensed to the industry. And these are the award, and the first award is given by the ministry, that is a Ghanaian Young Technology Innovation Award, and too many. And I first of all I acknowledge to my supervisor, Dr. Sudhir Pratap Singh, for giving me opportunity and trust me to I can do this job, and Chief Executive Officer for providing all kind of necessary things, faculty and technical staff, lab colleague, my family member, and a special thanks to financial support from CIAB, DBT, sir. Thank you so much.
Thank you, Dr. Patel, for your exemplary work on novel D-allulose 3-epimerase variants for the production of functional sugar and anti-diabetic and anti-obesity potential. Now the presentation is open for discussion. Any questions from the audience? Yes, sir, please.
So how, actually I'm a little confused that how you want to use this allulose or combination of 2 enzymes? Is it to add to the uses, or you want to convert these two fructose to allulose for use? Like, regular pills. What is the aim?
Sir, our aim is we first try this both the enzyme in any matrix. Then we treat it with the juices. After 1 hour reaction cycle, we can now remove the black enzyme, and
you can pass the juice through, remove it. Yeah. You will have the juice. It's fructose free, but it's still sweet.
Hello.
Have you done those experiments?
Yeah.
This one is like flow property of the column or something.
Yes, sir. We have to on small column in our lab. So we have
So the juice has pulp.
Yes, sir.
Pulp will block the column.
No. Juice is the first part we are doing the centrifuge the juice.
But that's not practical. So you have to think beyond it, application. That's why I was asking. So there is a juice which is always a juice will have pulp.
Yes, sir.
Whatever apple juice which we drink is not an apple juice. Every juice will have pulp, and you want to reduce the plastic equation. Yes. So you have to do experiment with the pulp.
But this is just an example. Basically, you want to do the most complete approach. We have some just example of this, we can this also. But our main aim is the production of the input.
That's what I'm asking. What is the aim? Is it to reduce fructose concentration of the juice, or to produce cellulose?
This is just an example we have.
Once again, thank you, Dr. Patel.
Thank you, Dr. Satyanarayan Patel. I request dignitaries to present memento to Dr. Satyanarayan Patel. Thank you, sir. Next speaker is Dr. Anita Ladwal.
Yeah.
Over to you, ma'am.
Yeah, our next speaker is Dr. Anita Ladwal. She is currently working as a lecturer, biology lecturer, and she did her PhD program from Guru Jameswar University of Science and Technology, Hisar. And during his area of research interest is celluloses, and she has published 6 publications from her PhD work, and she is having 1 patent also. So now I'd like to request Dr. Anita to present her on topic.
Thank you, sir.
All the best.
Very good afternoon, everyone. Before introducing myself, I would like to say thanks to you all, thanks to MI, thanks to the person present on the chair to give me such an opportunity to introduce myself here and present my topic before you all. And I would like a request to you all to give me 2 minutes for going outside of the scope of this conference just for the 2 minutes. Actually, presently, I am working as a biology lecturer in a school. And I taught students even of the 6th class as a science teacher. And in their book, 2 characters are given that are Bujo and Paheli. And Bujo is asking Paheli, let me speak in Hindi. Bujo is asking Paheli, कि Paheli, पर्यावरण का संरक्षण करने की जिम्मेदारी किसकी है? Then Paheli is saying, शायद सरकार की. And then next line is given, कि Bujo and Paheli will ask their teacher that the responsibility for saving this earth goes to whom? And then student asked me this question. Ma'am, who is responsible? Who is the key responsibility goes to whom? And I teach them that we all are responsible to save this environment. This responsibility goes to each and everyone. Each and everyone should take step in their daily life. Even every step we should take care. We should keep in mind that our key responsibility, our main responsibility in life is to save our earth. Actually, we just talk on this topic on Earth Day, maybe on Water Day. In our life, we all will get an opportunity to organize such programs like this conference, maybe marriage of our child, maybe another birthday party. So I request you all, please keep in mind in each and every function to save our earth. Thank you to all. The next, my topic is cellulose production from indigenous fungal strains. Why we are talking only about cellulases? Actually, cellulases are important enzyme both industrially as well as in natural world because they are having a capacity to degrade the most abundantly available agricultural biomass present on this earth. And after degrading this agricultural biomass, it gives us glucose, which can then be fermented into many commodity products. So it's important for many industries like food, animal feed processing industry, agricultural industry, pharmaceutical, starch industry, distillery and beverages, and so many more. A few more questions maybe rising in your mind. Are cellulose not available industrially? Yes, they are available in many of the companies like Enzyme Biotech, Zeneca Biotech Company, Enzyme Biosensors are synthesizing them. Then why there is a no need for synthesizing much more cellulose? Because these industrial enzymes are very costly to be used at large scale. Then how can we reduce the cost of these enzymes? We can reduce the cost of these enzymes by using cheaper substrate and potent synthesizers. And what can be these cheap substrates? These can be like agricultural waste and industrial waste. And this can be the potent producers. There are many sources which produce cellulases like bacteria, actinomycetes, but as per my knowledge, fungal cellulases are best cellulases because fungus cellulases, all the 3 complexes of the cellulases in their secreted form. And why agricultural waste? Because we all know India is an agricultural country, and as per the report of the MNRE in February 2019, 141 million tons of the surplus residue is generated, and out of this surplus residue, a very small amount is used as a fodder, and the left amount, about 92 million tons per year, is burnt as such in the field to clear the field for the next crop. And again, there are so many industries in India which flow their effluent as such in the rivers, which may be causing a severe problem of environment pollution, water pollution, land pollution. So by using this agricultural and industrial waste as an enzyme substrate, we can get rid of this environmental pollution, and also we will get a cheap substrate for the enzyme production. So where we go now? We will get a low-cost enzyme and get rid of so many environmental issues. For screening a potent producer, we have screened about 39 of the substrate collecting from various places, and from this substrate we isolated about 100 of the isolates. All the isolates were screened for cellulose enzyme production, and out of these, 5 best producers were selected. From these 5 best producers, we again screened them at higher level by DNS method, and the best producer named locally in our lab, NSF2 producer, was selected for further study. This NSF2 was again identified as Aspergillus niger from the IARI New Delhi. Various substrates selected for the study were raw wheat straw, rice straw, maize leaves, maize cob, rice bran, and the list for industrial waste which we screen our study are given. And at last, maximum enzyme production was found by wheat straw moistened with the whey as a moistening agent, which were used in our study. By the enzyme produced by our local isolate, NSF2 fungal isolate, we employed our enzyme in de-inking of the paper. De-inking of the waste paper. For the de-inking, firstly we prepared some handmade sheets by using a mold which was locally designed in our lab. These 4 types of paper were produced in our lab. A for the control, which was not pre-treated. B for the Na was pre-treated. C for the sodium silicate pre-treated. And D for pre-treatment after our enzyme for de-inking. All the sheets were characterized from Saharanpur in terms of brightness, brush index, tear index, tensile strength, residual strength. And after studying these, we found that the cellulose was surprisingly giving very good results. After these studies, we also characterize our seed by sun micrographs. Under sun micrographs also, we found that our cellulose enzyme was giving best result for de-inking, even as compared to sodium silicate, which is employed even industrially. Again, the seeds were screened under fluorescence micrograph because all the dyes containing some fluorescence compounds. No one other have reported the fluorescence micrograph as an evaluation method for de-inking. It was novel in our study, and we have been granted a patent for this also. FTIR spectroscopy was also employed to support our results, and again under FTIR spectroscopy we found that the cellulose enzyme was giving best result for de-inking. Our results were also supported by fluorescence spectrophotometer study. Thus, finally, I can conclude that for de-inking, in place of using chemicals, we can use cellulose enzyme, which is giving very good results, and the techniques employed for the evaluation of de-inking used in our study were not using any chemicals for any time for the sample preparation, which will save your time and also the capital cost. These are various publications, and this is one of the patent which is filed for the de-inking efficiency measurement. Those are various workshops and conferences attended by me. Thank you everyone for listening so patiently. Any questions, please?
Yeah, congratulations, Dr. Anita, for
Thank
your
you, sir.
excellent presentation as well as your publications and patenting. The session is open for discussion. Any couple of questions can be asked.
I think there is no one. Thank you everyone again.
Thank you very much.
Thank you, ma'am. Thank you, ma'am. And I request dignitaries to present a token of memento to our presenter. Thank you, Dr. Anitha Devi. And the next presenter is Mr. Yogesh Chandra.
Our next young scientist awardee is Mr. Yogesh Chandra. He is currently working as a PhD scholar at Guru Jambeshwar University, Haryana. He already published 15 research articles and received a couple of best poster and young scientist award. So with this brief introductions, now I request Yogesh Chandra to present his research finding. Yogesh, please.
Good evening, everyone. Now I'm going to present my talk on resistance evolution against host-directed antiviral agents, where Buffalopox virus switches to use a host factor P38 gamma instead of P38 alpha under long-term selective pressure of an inhibitor targeting P38 alpha. So I'm starting with a general introduction about virus. Buffalopox disease caused by a member of Poxviridae family virus, Buffalopox, and this is a emerging zoonotic disease. The clinical signs of BPH infection in animals like development of poke lesion and the other, and in humans the development of poke lesion and all over the body. And BPXV is prevalent in India, Egypt, Turkey, Indonesia, and some African countries. So currently there is no approved antiviral to treat BPXV infection. So there are two approaches to develop antiviral drugs against the viral infections. First is to directly target viral proteins, and this comes with a disadvantage of the acquisition of drug resistance. Now we are working on a novel strategy where we are targeting host factors instead of targeting viral proteins. Because virus is an obligate intracellular parasite and it requires more than 1,000 proteins for infection, and it critically relies on host machinery for its effective replication. In this instance, we screened a library of kinase inhibitor and we got a potential hit against BPXV that gave us a clue that this P38 MAPK may support BPXV replication. Next, we generated P38 alpha and gamma knockout cell lines by using CRISPR-Cas9 mediated genome editing, where we transfected virus cells with guide RNA targeting P38 alpha and P38 gamma. The transfected cells were sorted by using FACS, and the single cell clone was scaled up by serial dilution, and the generation of P38 alpha and gamma knockout cell lines was validated by amplification of targeted region of P38 alpha by PCR, and following by the nucleotide sequencing and Western blot analysis, where we can see that P38 alpha expression is completely absent in knockout cell lines. Similarly, P38 gamma was also validated by nucleotide sequencing and Western blot analysis, where we can see that the P38 gamma is completely absent in P38 gamma knockout cell lines. So next, we evaluated that and we can see that in dose dependent manner, as well as the infection of virus in P38 alpha knockout cell lines impairs the BPXV replication, which confirms that P38 MAPK supports BPXV replication. Next, we were interested to know that which step of virus life cycle is affected by inhibition of P38 alpha. So we performed the experiment and we found that the treatment of cells with P38 MAPK inhibitor reduces the BPXC protein synthesis as compared to control. The inhibition of or reduction of BPXC protein synthesis, which in turn reflects in the reduction in BPXC DNA level. So it is confirmed that P38 MAPK supports BPXC protein synthesis. Next, how this P38 MAPK support BPXC protein synthesis, we evaluated the activation kinetics of P38 upon BPXC infection, and we found that in initial hours and the later hours of infection cycle, P38 alpha got activated, where the phosphorylation is the marker of activation. And interestingly, this is EIA4E, which is a host, which is a member of host translation machinery. The activation kinetics of EIA4E is overlapped with the kinetics of P38 alpha. Next, we investigated that we treated the cells with P38 inhibitor, and we found that P38 inhibitors blocks the activation of P38 alpha, and similarly the less blocking of P38 activation impairs the activation of EIA4E, which suggesting that there is an P38 and EIA4E signaling may support supporting BPXC protein synthesis. Next, P38 MNK and EIA4E signaling pathway is involved in the BPXC protein synthesis. So we treated the cells with MNK1 inhibitor and EIA4E inhibitor, and we found that the treatment of cells with MNK1 inhibitor reduces the BPXC replication, as well as the treatment of cells with EIA4E inhibitor reduces the BPXC replication. This EIA4E actually a member of translational machinery of host. EIA4E confirms the integrity of mRNA. Before translation, it interacts with another partner, that is EIA4G, and which this complex confirms the integrity of mRNA. If mRNA is complete, then translation starts. So we next investigated whether the treatment P38 inhibitor affects the interaction of EIA4E and viral mRNA, and we found that the treatment of inhibitor reduces the interaction of viral RNA with this host factor. So we concluded that P38 MNK and EIA4E signaling axis is supporting BPXC mRNA translation. Next, in my first slide, I said that this is a novel strategy because we are targeting host factors instead of targeting viral factors. So we investigated that in case of P38 inhibitor, the virus acquires resistance or not, and so we passaged the virus 60 times in the presence of drug, and we evaluated the sensitivity of passaged virus, control passage virus, and P0 virus, which was not passaged in the cells. And we found that the drug inhibitor passage virus was less sensitive as compared to P0 and control passage virus. The similar result were observed in the P38 alpha knockout vivo cells, which confirms that upon long-term passage under the selection pressure, the virus acquires resistance, even in the presence, even if we are targeting host factor. Next, we dissect how this virus replicates even in the presence of a inhibitor which is targeting, which is blocking the host factor. We evaluated sensitivity of P0 virus, control virus, and P60 virus, P60 inhibitor virus in P38 alpha knockout cell line and P38 gamma cell lines. As we can see in case of P0 virus, the sensitivity, the fold suppression or sensitivity was higher as compared to P38 gamma cell lines. We suggested that P0 virus relies on P38 alpha initially. Similar results were seen in P38 control virus, but virus also dependent on P38 alpha for its replication. But in case of inhibitor passage virus was more suppressed in P38 gamma knockout cell line as compared to P38 alpha knockout cell lines. This is suggesting that this inhibitor passage virus switches to use P38 gamma instead of P38 alpha under the long-term selection pressure of inhibitor. Next, we went for the sequencing of virus, and we found that there are two unique mutations in ribonucleotide diphosphate reductase and promoter region of serine and threonine kinase in the genome of resistant virus variant. This is the signaling axis which regulates translation of BPXA proteins. So first, when P38 get activated, it activates MNK1. Upon activation of MNK1 activates EIA4E, and the translation goes on. If you are blocking these host factors P38 by inhibitor, if you are blocking MNK1 by this inhibitor, and if you are blocking EIA4E, it reflects in the reduction in viral protein synthesis. So this signaling axis regulates BPXA protein synthesis. Next, in our case, we observed that over the long-term selection pressure, BPXA acquires resistance. So this is the graphical view that upon long-term selection pressure, BPXA utilized P38 gamma instead of P38 alpha. In conclusion, this is the first report where we are showing that P38 MAPK host factor supports BPXA replication. And these host dependency factors may be targeted for the development of antiviral drug development. Actually, BPXA does not select, BPXA easily not select P38 inhibitor resistant mutant, but upon long-term selection pressure of a drug allows virus to switch to use alternate host factor. This is the first evidence where a virus was shown to bypass host dependency in the whole world. For the first time we reported that how much virus is the smartness of the virus, and we clearly mentioned the switched host factor or alternate host factor. For the first time we have this described in the world. This paper is published in Molecular Biology and Evolution. The journal is have a 5-year impact factor 20 and published by Oxford University Press. So during my career of 5-6 years, I have published more than 50 research papers, and I isolated 100 different viruses from outbreak and different infected samples. I published in my papers in antiviral research with the impact factor of 10, molecular biology and evolution, frontiers in microbiology. One of my reviews in medical virology. Recently, my paper is accepted in Nature Scientific Report. So, and moreover, Encovax, this is a vaccine for animals against SARS-CoV-2 was developed by and patent has been filed. And this vaccine is released by Central Agriculture Ministry in New Delhi. The technology is transferred to Biovet India Private Limited, Bengaluru. the lumpy Provac vaccine. This is another vaccine developed by me, and which was released by Central Agriculture Minister 9th August in this year in Krishi Bhavan, New Delhi. This technology transferred to Biotic India Private Limited, Bangalore, in 81 lakhs. I would like to thank a lot of thanks to my supervisor, Professor Nida Singh, from JJ Hisar, Haryana, and Dr. Naveen Kumar, my supervisor at NRC Hisar, and Dr. Sanjay, Dr. VST, these are all my supervisors. And thanks to you, thanks to AMI to consider my work for this award.
Thank you, Dr. Yogesh, for your excellent research on the evolution of resistance against buffalo pox virus. And congratulations once again for getting this prestigious Young Scientist Award. Now the presentation is open for discussion. We can quickly take 1 or 2 questions. Any questions? Yeah, fine. If there is no questions, then congratulations once again. Okay, thank you.
Sir.
Questions? Okay, please, sir.
Yeah, I was actually at this initial part. You taught the mechanism of resistance, the sensitive virus. But what type of mechanism of resistance you
Sir, actually, we are blocking a host factor P38 alpha. So the P38 alpha is initially supporting viral protein translation. If we are blocking with a inhibitor, the viral replication goes down. Yes, sir.
target modification?
No. This is just the blocking of host factor by an inhibitor.
So target modification.
Sir, I didn't get what you are.
What is the mechanism of resistance?
Sir, mechanism of resistance acquisition is the switching to use another host factor in the absence of that host factor. This is the mechanism. We have reported for the first time.
Thank you, Mr. Yogesh. Thank you, sir. And the next presenter is Dr. Vikas Dul. Dr. Vikas Dul, it's over to you.
Yeah, our next young scientist awardee is Dr. Vikas Dul. His current designation, currently he's working as an assistant professor in Dayanand University, Rohtak, Haryana. And he did BTech and MTech. He is a fascinating scientist working on biosensor technology, nanomaterial synthesis and diagnostics. And he has published nearly 19 research articles with a fascinating number of citations as well as H-index and I-index. And he received best research paper awards in different international and international conferences. And he's a fellow of DST Inspire for JRF as well as SRF. And he got a CSR JRF also. So with this brief biodata, I would like to welcome Dr. Vikas Dul for his presentation.
Good afternoon, everyone. And thank you, sir, for such a nice introduction. And my topic of presentation will be the fabrication of nanomaterial-based biosensors for detection of acutely toxic organophosphate compounds. Now, what are organophosphate compounds? These compounds are basically present in the pesticides which we regularly use in agriculture. As we all know that the population is exponentially growing day by day, so there is a very intense pressure on agriculture to support the growing population. So that's why the farmers and we all are using the pesticides in large quantities without knowing the composition. So this is finally contaminates our environment and also the microorganisms which are present in the soil, which are very helpful for the growth of plants. So here we these OP pesticides are the neurotoxics and inhibits acetylcholine esterase enzymes, which is a very necessary enzymes which break down acetylcholine to thiacolines and the other compounds which acts as the neurotransmitters in our body for the signaling purpose. Eventually, their inhibition results in the malfunctioning of the central nervous system, interferes in muscular responses, and eventually the death. OP presence is expected in water resources, soil, fruits, vegetables, and in the recent studies, it has also been reported that it has found OP compounds are present even in the mother's milk. So that's why this is a very serious issue. So commonly used OP pesticides have included parathion, paraoxones, and diazones, and methylparathion, and other compounds. There are a lot of methods available. These are conventional methods for detection, successful detection of these compounds, such as colorimetry method is there, high pressure liquid chromatography is there, gas chromatography, capillary electroscopy, and so on. But these methods are inefficient in detecting the lower amount of organophosphate compounds present in the samples or present in the real samples, you can say. So these conventional laboratory methods are also labor intensive and they cannot test the sample on spot. So they are not for the on-site detection. So that's why these limitations of these conventional methods can be overcome by using the nanomaterials. So that's why we have fabricated nanomaterial based sensors to detect the compound. So how a biosensor works? Basically, biosensor is a 3-electron system in which we use a support to immobilize an enzyme that that detects the sample, that detects any physiochemical properties, physiochemical changes taking place in the reaction mixture in which we have put the particular electrode. And the transducer may contain the biorecognition element. It may be enzyme or it may be any antibody can be used there. So this is the basic assembly of the electrochemical biosensor. For this work, we have used the gold nanoparticles. We have synthesized the gold nanoparticles in our laboratory by following Golay and Morphy et al. With some modifications. After that, we characterized the nanoparticles prepared in the laboratory using several techniques. So for characterization of nanomaterials, we go from UV visible spectroscopy to transmission electron microscopy. After that, we prepared an electrode in our laboratory comprising of various nanomaterials, which includes the carbon nanotubes and the nanoparticles. Finally, we got that core electrode, the basic electrode which we developed using the mixture of multi-walled carbon nanotubes and the gold nanoparticles. We call it as a core electrode. On that core electrode, we deposit the mixture of enzyme. We try to deposit or immobilize the enzyme covalently on that so that the electrode can be reused, as enzymes are very costly and the cost of purification and production is very high. So we emphasize on the covalent immobilization of the particular enzyme. Finally, we get the working electrode in which we have coated that electrode with Nafion. This is the final working electrode, and these are the synthesis method. The synthesis of nanomaterials is very interesting, and we can synthesize the nanomaterials within 5 to 30 minutes. So it looks very easy that we can synthesize any nanoparticle easily, but it is very tough. You can get it in the coming slides. So for synthesis of the gold nanoparticles, we use the gold chlorous acid and sodium citrate as the reducing agent. When we use these chemicals for the chemical synthesis of the gold nanoparticles, within 2 to 3 minutes, we will see or detect the change in color from within 2 to 3 minutes from the transparent to the wine red color, or you can say the brick color. And we continue this to 4 to 5 minutes so that we can get the stable nanoparticles. There must be some stability. So finally solution was centrifuged and the pellets were collected and dried, and these are the nanoparticles, gold nanoparticles, which we have used in the further study. For confirming the successful synthesis of nanoparticle, firstly we can use the colorimetric detection, as I have earlier discussed that the change in color will be the first confirmation to the synthesis of the particles, but it is not the synthesis of the, we cannot say by seeing the color that these are the nanoparticles. Only there is a possibility that the material which we have synthesized is the compound, that particular compound. If we are synthesizing gold, then it will be the gold. We can tell it from the color. Okay. After UV visible spectroscopy, every compound has its own absorbance peak, and the gold has this absorbance peak at 540 nanometer, as we can see from this graph. After that, before moving to the further experiment, we have to confirm that the particles which we have synthesized are in nano range or not. So for this, we use the technique particle size analyzer, which tells us about the average size of the nanomaterial or the nanoparticles which we have synthesized in our laboratory. So from these results, we can see that the nanoparticles which we get in the particular study are to 10 to 40 nanometer. Below 100 nanometer, the properties of above 100 nanometer, the properties of nanoparticles are of no use. So we focus on that. The nanomaterial size of nanomaterial will be less than 50 nanometer. After that, one more confirmation, characterization of the nanomaterial which we have synthesized is X-ray diffraction, which is basically for looking for the crystalline structure of the nanomaterial which we have synthesized. From these, we can see the crystalline peaks at different angles, and these peaks are matched with the JCPDS database. You can see this is Joint Committee for the Powder Diffraction Standards. From that, we compare our results, and if the peaks match, we can go further for the experiment. These are the transmission electron microscopic images of the nanoparticles which we synthesized in our laboratory, and transmission electron microscopy gets the exact shape and size of the nanomaterial which we have synthesized. But the particle size analyzer only tells us about the average size range of the nanomaterial. Now we have to prepare, we prepared core electrode, working electrode by using multi-walled carbon nanotubes and a composite mixture of multi-walled carbon nanotubes and gold nanoparticles which were pasted on the gold wire. We can use any other wire also, but in this study we use the gold wire to get the proper results or to achieve the highest electrocatalytic activity. For that, we have taken a tube which is 1 centimeter long and 4 millimeter in diameter and filled the paste of multi-walled carbon nanotubes and gold nanoparticles in that by mixing it in the paraffin oil to get it the consistent mixture. After drying, we removed the tube and we put the carboxylated single-walled carbon nanotubes and our enzyme acetylcholine esterase, which has been taken from the microorganisms, or the mixture was casted on the surface of electrode core. The core will be the multi-walled carbon nanotubes comprised of and the gold nanoparticles on the gold wire. Finally, after immobilizing enzyme, we use the Nafion layer. Nafion is basically a polymer which helps in preventing enzyme from leaching and membrane fouling so that it can be reused. So these are the characterization images of the working electrode. The figure A, in figure A, this is the bare gold electrode. This is like a blank photograph. This is of bare electrode. The figure B is comprises of multi-walled carbon nanotubes and the gold nanoparticles. And finally, this is the figure C. The image C tells us that this is the final image coated with Nafion and having immobilized enzyme. So we also characterize this electrode using FTIR, Fourier transform infrared spectroscopy, to know that if our enzyme has been covalently immobilized or not, and we have got the good peaks which shows that it has been covalently immobilized. Now, after this development of the electrode, final electrode, we move to the electrochemical studies of the particular biosensor. In electrochemical studies, we optimize it at optimum temperature, working temperature, optimum, what is the optimum pH, and what will be the substrate concentration to get the optimum results. So the equilibrium of the equilibration of working electrode was done at 0.4 volt before each run. This is only to get the steady state, and biosensor was tested at 0.4 volt. Substrate acetylthiocholine iodide was added into the electrochemical cell. Basically, electrochemical cell is the cell having the reaction mixture along with the buffers. So ATCI was hydrolyzed enzymatically to thiocholine. Further, thiocholine was oxidized versus these AgCl reference electrode. This is basically
sorry, yeah. 4 minutes.
2 minutes. Okay, sir. So we have this is the reaction scheme, and after that, these are the cyclic voltammetric studies. On cyclic voltammetric studies, we optimize our electrode at the lowest current so that the species can be generated, and the amount of current generated is inversely proportional to the amount of organophosphate compound present in our sample. So the amount of current generated will be inversely proportional to the amount of pesticide, or the, you can say, OP compound present in our sample. In kinetic properties, the response current changes slightly between pH 5 to 7. So the current, so maximum current was observed at pH 7. That's why we used this pH. Optimum temperature was 30 degrees Celsius, and every time when we use it, we scan the electrode from 0.4 volt to 0.6 volt. This is the effect of substrate concentration. The biosensor showed a hyperbolic relationship between the activity and substrate concentration up to 600 micromolar. So we have used different pesticides or organophosphate compounds such as methylparathione, monocroptus, and chlorophyresis and endosulfan, which are generally present in our pesticides and affect the environment along with the microorganism. The inhibition effect of immobilized enzyme is directly proportional to incubation time, unless it has reached to saturation level. After 10 minutes, less decrease was observed. So 10 minutes was selected as the incubation time. So these are the linearity and detection limit, which show that our developed sensor is properly linear and a good linearity range, and it has also been tested for the percent analytical recoveries with good accuracy, and reproducibility of the electrode was also good. We have also correlated our electrode with the presently conventional methods such as HPLC. A good correlation has been achieved. Interference studies, we studies different species which are present. So we observed on 7 to 9% of interference from glucose, fructose, and sucrose. It can also be, electrode can be regenerated only by changing the pH. So we have tested in spiked water samples, testing milk samples, and in these vegetable samples, and we get good analytical recoveries. Soil samples, this is the reusability. We can use it at for 2 months. It is 30% more activity. Now our this research has been compared with earlier reported methods. So in conclusion, the new fabrication strategies for development of working electrode for on-site monitoring. Its best is it is use can be on-site, and also it is not labor intensive. No reaction mixture is prepared. Okay, sorry, no sample preparation is required. So that is my work, and thanks. If any questions, you can surely ask.
Yeah, thank you very much.
Thank you, sir.
Very nice presentation, very nicely presented.
Thank you, sir.
On this, especially we facing a lot of issues on organophosphates.
Yeah.
What's
Yes.
the advantage of this biosensor?
Yes, sir. Biosensors.
Good presentation. Yeah.
Thank you, sir. Thank you.
The topic is open for the discussion. Any queries? Anyone from the audience side? Yeah, if no one is there, congratulations.
Thank you, sir.
Thank you very much.
Thank you.
Thank you, Dr. Vikas Dul. Yes. Our last presenter is Dr. Junaid Jibran Javed. Over to you, sir.
Okay, thank you. Our last speaker in this lecture, in Scientist Lecture Award series, is Dr. Junaid Jibran Javed. He is currently affiliated as an assistant professor, Institute of Health Science, Presidency University, Kolkata. He did his PhD in microbiology from Bose Institute, Kolkata. His research interest is in drug resistance, immunology, and infectious diseases. He has already published 21 research articles in leading international journals, and he is an editorial member of prestigious journals, including Frontiers in Cellular and Infection Microbiology. He has been bestowed with the Neelarattan Sirkar Prize. Okay. And then Professor Asima Chatterjee Young Scientist Award. And he has already sequenced 15 novel Mixo bacterial strains during his PhD. And Dr. Junaid, please go ahead.
Thank you very much, sir, for the nice introduction, and a very good afternoon to all the audience present. So my topic of today is combating protozoan infection with the immune cells, a novel strategy to overcome the drug resistance. So before going to the greater detail, I would like to give you a brief history of the medicinal perspective, introduction to the disease model with which I have worked, and the proposed model and the result and discussion associated with it. So as we know from the very initial days, human civilization has started practicing the medicines through healing their wound by covering the wound with the help of plant parts. Later on, gradually they have learned to use the plant extract, not only extract to allow as an ointment above the wound, but as also oral medications to boost their immune responses. These things also documented into the very old scriptures, Eber's Papyrus, which shows that more than 200 diseases and the use of various types of plant extract that has been used in the very early days. Gradually, people have learned how to use formulation-based medicines, and then we have entered into the laboratory medicines. These laboratory medicines later on explored the avenues to use this formula-based medicines, and we have also come across that how to do the group modification to come up with more effective types of medications, whether it is antibiotics, whether it is any kind of a certain kind of infections. Finally, we have entered into the era, this is called vaccination, where the infection which will happen in future we can protect itself. So we know that this person, Edward Jenner, he is a father of immunology, and he has accredited as the person who has saved more life on earth than any other person's contributions. So this particular slide is showing that Steve Parker has written this book that shows that human has a ever compelling interest to find the cure of all the diseases. Keeping this quest in mind, we have come across the use of these cells of the immune system to cure the infectious disease. For that, we have selected this leishmaniasis. So leishmania is a very old disease. It is happen with the help of with the infection of protozoan parasite that is belonging to the genus Leishmania, and it is transmitted by the bite of the infected sandfly. On the basis of the different association of different kinds of parasite, it is actually of three types, like cutaneous, mucocutaneous, and visceral form. The visceral form is also known as Kala Azar, which is caused by Leishmania donovani, Leishmania infantum, Leishmania chagashi. So it is the most fatal form, as it affects the visceral organs of the human. The common signs and symptoms includes the fever, weight loss, enlargement of organs, due to which the abdominal swellings take place, anemia and darkening of skin. Due to this fever and darkening of skin, this disease is also known as Kala Azar or black fever in India. The mode of infection, as we know, that the female sandfly during their blood meal infects the mammalian host, and later on this parasite enter into the macrophage systems where it infects more macrophages, and later on the organ parasite burden increases, leading to the abdominal organomegaly. This is the current statistics that is associated with the Kala Azar. And if we deal with the host versus parasite scenario, we can see that when infection happens, the kind of immune response like pro-inflammatory cytokines becomes very high, that cell-mediated immunity is compromised, whereas when there is an effective immunity, there is good amount of T cell subsets in our body that helps in the protection from the disease. So in this case, our objective is how to use which particular types of immune cells will be the suitable candidate, and how to use these immune cells. For that, we have proposed a model that when the antigen presenting cells, as the parasite infect the antigen presenting cells, if it is stimulated with any pathogen recognition receptor, it secretes some factors. These factors actually convert the T cells into different subsets of the T cells, and then these T cells become the source of the cytokines which give us the immunity to fight against the disease. Keeping this model in mind, we have isolated the bone marrow derived dendritic cells. We have stimulated the dendritic cell with soluble leishmanial antigen SLA and peptidoglycan, so that the dendritic cell will get polarized against the parasite and it will get a pro-informed response. We had adaptively transferred it, and later on we have given the infection to the mice, and we have checked the purity of the dendritic cells and the maturation markers after the SLA-PGN dual stimulation. And we have found that the maturation markers like MSC class 2, CD80 and CD86 were significantly increased due to this SLA-PGN stimulation. Later on, we have used it for the infection model. This is the way we count the intracellular parasite, and we found that the organ parasite burden was significantly reduced in the SLA-PGN dually stimulated vaccinated animals. When this actually significantly reduced, we wanted to know what is the status of the different cytokines in the blood of this animal. So we found that it was among the various cytokines, the expression of IL-17 was significantly increased. This is one of the special types of cytokines which is secreted when the dendritic cells or any antigen presenting cells is stimulated and secretes some factors which convert CD4 plus to TS17 cells, and then these TS17 cells become the source of this IL-17. So we wanted to know what are these factors and how this helps in the conversion of this CD4 to TS17 cells. So we have found that through flow cytometry experiment, during infection, the IL-17 positive CD4 plus T cells that expresses ROR gamma T, that is very specific for TS17 cells, decrease, which was increased significantly after SLA-PGN based dendritic cell vaccination. And the associated cytokines like IL-6, IL-23 was also increased with the dual stimulation. Now we wanted to mimic the same condition in the in vitro setting, and we have performed the co-culture experiments, and we found that when dendritic cell is co-cultured with CD4 plus T cells stimulated with SLA and PGN, it increases the expression of certain factors which convert this CD4 plus T cell to TS17 cells. And when we have neutralized this IL-6, IL-23 and TGF-beta, the expression of IL-17 or the population of IL-17 positive CD4 plus T cells were significantly down. So this is proving that for after the stimulation, these are the factors IL-6, IL-23 and TGF-beta that actually responsible for the conversion of CD4 to CS17 cells. So what is the role of other signaling molecule inside the dendritic cells in this scenario? As far as the text are concerned, various protein kinases are there which helps in the regulation, lineage commitment and proliferation of these Th17 cells. So for that we have focused on the different isotypes of protein kinase C. We have found that this expression of this protein kinase C was significantly increased, specifically for the isoforms of PKC alpha and PKC beta. Among these PKCs, which PKCs involved in the more prominent role for this Th17 cells generation, for that we have performed this silencing assay, and we found that silencing PKC beta specifically downregulated the expression of IL-6 and IL-23, which are the two cytokines responsible for the lineage commitment of the Th17 cells and the proliferation of these Th17 cells. And the population of Th17 cells were also compromised after this silencing this PKC beta. So this proves that the role of PKC beta in this Th17 cell generation. Finally, we wanted to know the role of IL-17 in the protection against this parasitic infection. And when we have introduced the IL-17 neutralizing antibody, the parasite burdens were profoundly increased inside the mice. So for, this can be summarized as when the dendritic cell is stimulated with SLA and PGN, it increases the maturation marker like MSC class 2, CD80 and CD86, and in one way it is increasing the expression of PKC beta, which increases the expression of polarizing cytokines which convert CD4 to Th17 cells. That becomes the source of IL-17, and it actually effectively reduces this parasite burden. So this work has been accepted in FIMS pathogen and disease. Apart from that, we have also worked for identification of the role of different NOD-like receptors in Leishmanian pneumonia infection, and that we have found that the role of the parasite exploited the NOD2 inflammatory pathway. So this work is published in Biomedicine and Pharmacotherapy. In addition, we have also explored the role of different transcription factors related to the antigen presentation and cross presentation during infection. Recently, this work has been accepted in experimental parasitology. Apart from that, in addition, we are also working with some proteins of the Microbacterium origin, and we found that this MPT63 can mimic somewhat like toxin-like manner during the infection. All this work, I would like to acknowledge the funding agencies, specifically CSIR, and currently my lab is getting funded from ICMR and UGC. I would like to convey my heartfelt thanks to my mentor, my PhD guide, Professor Subrata Majumdar, and my collaborator, Professor Krishnananda Chattopadhyay from Indian Institute of Chemical Biology, and the place which is associated currently I'm associated with, that is Presidency University. Thank you very much. Thank you very much for the patience.
Thank you. Thank you, Dr. Junaid, for your excellent presentation on exploring immune cell for combating various protozoan diseases.
Thank you very much.
Congratulations once again for getting this prestigious scientist award. Now the presentation is open for discussion. We can take one or two questions quickly.
So I don't think there is any question.
There is no question. Once again, thanks a lot.
Thank you very much.
And in fact, we congratulate all young scientist awardees, Dr. Angul Vaishnav, Dr. Gaurav Sharma, Dr. Satya Narayan Patel, Dr. Anida, Dr. Yogesh, Dr. Vikas, Dr. Junaid, for your excellent work. And we urge all of you to please keep up with this good work, this scientific temperament. And we wish you all the best in your future endeavor. Thank you so much.
Thank you very much, sir.
Thank you.
Thank you, sir. Thank you for your wonderful presentation. Now we come to the end of the session. I request Professor V.K. Chaudhary, sir, to honor the dignitaries in the dais. Please, sir. Thank you, Professor P.C. Abhilash, Banaras Hindu University, Varanasi, for chairing this session, and Dr. Prakash G, IARI, New Delhi, and Dr. Rashmi KC, University of Mysore. Thank you all for your presence. Thank you very much, sir. Now we came to the end of the session. I thank each and everyone for a patience hearing and more than a valuable presence. Thank you very much. And the refreshments are arranged outside. Please have. And general body meeting will be held in the same hall at 6 p.m. All the AMI members are requested to present here at 6 p.m.
ಅಲ್ಲಿ ಎಡಿಟ್ ಮಾಡ್ಕೊಂಡು ಹಾಕ್ಬಿಡ್ತೀನಿ ಇನ್ನೊಂದು ಫೋಟೋ ಹಾಕೊಂಡ್ಬಿಡ್ತೀನಿ ಸ್ಮೈಲ್ ಮಾಡ್ದು ಸೀರಿಯಸ್ ಆಗಿರ್ಬೇಕಾ ಎಡಿಟ್ ಮಾಡಿ ಇಮೇಜ್ ಹಾಕ್ಬಿಡಿ ಎಡಿಟಿಂಗ್ ಅಲ್ಲಿ ವೈಡಲ್ ಇದ್ದಾಗ ಫೋಟೋಗೆ ಈ ಪ್ರೋಗ್ರಾಮ್ ಗೊತ್ತು ಸಂಬಂಧನೇ ಇತ್ತು ಸಂಬಂಧ ನೋಡೋಣ ಈ ಬೈಟಲ್ಲಿ ಫೋಟೋ ಹಾಕ್ತಿಲ್ಲ ಫೋಟೋ ನೋಡ್ಬಿಟ್ಟು ಬೇರೆಲ್ಲ ಓಕೆ